you. All right, everybody, thank you for coming back in a relatively timely manner. I think we managed to catch up most of the delay, which is good. We're going to get started with the second ecotoxicology session. So as you noticed this morning, the first ecotoxicology session was largely oriented towards heavy metals and other chemical pollutants. This ecotoxicology section has some other types of chemical pollutants, not heavy metals, and largely plastics, microplastics. And so our very first speaker, I'm very happy to welcome Dr. Gisela Poletta from Argentina.
Hi, everybody. Thank you for being here. Okay, I'm going to present some results of a study called Emerging Contaminants in Wetlands, the effects of anti-parasitic drugs in Caima latirostris. This is the work of my postdoc student, but well, I'm here, so it's my responsibility. So, well, you know, in Argentina, as in many other traditional agricultural countries, the land dedicated to crops has has dramatically increased during the last 3 decades. And as you can see in the pictures or in the right, that has to do with the implementation of a technological system that includes the use of, well, genetically modified seeds that can tolerate a lot of environmental stressors, such as drugs and for example, coal. And as well as the increasing use of pesticides during the entire moment of the growing. I mean, the entire period of growing of the crop. So that means that the frontiers, agricultural frontiers in the country had spread a lot, and in Argentina particularly, that's made that this area overlap with the geographic distribution of the 2 species of caiman that we have there, that is Caiman latirostris and Caiman jacare. As a consequence of this change in the use of land, the system of livestock, of raising livestock also changed, and has led to the more intensive kind of system. I mean, it changed from a traditional pasture system to the intensive or semi-intensive system that is known as feedlots. And so the problem that this kind of system has, that is that there is a lot of, a higher number of animals per area, so that contributes to the increase of illness in the animals, and the requirements of the use of many antiparasitic drugs, or the increasing use of antiparasitic drugs, that also contribute to contamination of the environment. So in this slide, you can see that the main antiparasitic drugs are used for in Argentina, are fenbendazole, albendazole, and ivermectin. And that are eliminated through urine and feces, and that can lead to the water bodies that are near these kind of places. So there are a higher risk for our species too to be contaminated by this kind of emerging compounds. We have been, during the last 20 years, investigating the effects of pesticides in our country. And so our main concern is what can be happening with when these kind of compounds that are in the same environments acting together as mixtures can have in the different stages of the species. So, well, these are two images of the main, water resources related to the species in Argentina, the Salado River and the Paraná River. And these are levels of antiparasites that were determined in recent works. So as you can see, there is a level that is worrying, and there is only one study made in reptiles around the world on the effects of antiparasites, and is in the species Eremias argus, exposed during 30 days to abamectin. And well, the authors report problems or alteration in oxidative stress, locomotion, and changes in behavior related to predation. So well, in our place, we can see in natural population or environments images like this. This is, I don't have a, but this is a place where you can see there is a soybean crop. There is a soybean crop. It's the same place, taken from different, points. This is soybean crops. This is an intensive livestock, and this is a small lagoon where a female, or maybe more than one female and a male surely, are staying, because we find there. Well, you can see here is very small there. But this is a nest, a caiman nest there, where we found eggs. And so, as I said, our worry was to know what could be happening with these populations that are in very modified environments. We know from our previous studies that a pesticide can alterate all these parameters that had to do with oxidative stress, antioxidant defenses, genotoxicity, both in terms of structural and functional, for example, in relation to gene expression, and well, so our aim was to try to evaluate the same parameters in the most used antiparasites. So, well, this was a subchronic site during 2 months with caimans, juvenile caimans of 6 months, coming from 4 different clutches from an area without apparent contamination, that is a natural reserve in Argentina. We use a very low concentration comparing with those that were found in the environments in the studies I mentioned, and the nominal concentration determination made by HPLC show a recovery percentage of more than 95%. Well, so the parameters include were growth during that period, genotoxicity, oxidative stress, corticosterone levels, metabolic parameters, hepatic enzymes, and gene expression. I'm going a little fast. We found that both pembendazole and ivermectin induce lower growth in the animals during this period. And especially you can see that ivermectin has almost, more than less than the half of the negative control. When we analyzed the comer assay, we saw that ivermectin produced DNA damage, but not the benzimidazoles, and no one of them induced nuclear abnormalities. We found both for fenbendazole and ivermectin lipid peroxidation, that is, it can be related to an effect on cell membranes. We don't have effects on gene expression, but we can see that for superoxide dismutase, we have a tendency that maybe is not significant because of data dispersion. We found metabolic alteration both in triglycerides and glucose, and so that maybe has a consequence in energetic parameters. And in the hepatic enzymes, we only found effects on AST for, again, fenbendazole and ivermectin, and similar effect in corticosterone that the data was reported this morning by Jeremy. We found a lower corticosterone level in fenbendazole that can be maybe related as inhibition of the HPA stress response. Well, as some concluding remarks, we can see that subchronic exposure to low concentration of antiparasites induce measurable genotoxicity, oxidative stress, metabolic alterations in these species, and especially growth, small growth of the animals during the period. Of course, it is necessary to continue studying this parasite because antiparasites, because the effects are quite, complicated, are very, in some cases, we expect to have a rise, for example, in corticosterone, and we found lower values. And well, the important is this is the first report on the toxicity of antiparasites in non-crocodilian species, and it's really made us have more concerns about the natural population that are living in this so modified area. So, well, just to thank you, all of you, for being here, and well, the supporting agencies, and this is my email, and you can find me there.
Thank you very much, Gisela. If I can have Joey Brown to the front, please. Our next speaker is Joey Brown from Jamaica, Hope Zoo.
All right, thank you, Matt. Yeah, so we're going to be talking about plastic ingestion and some crocs in Jamaica, using necropsy exams over the last few years to document an emerging conservation threat. So briefly, I just want to give you a background. Distribution and habitat plays an important role. So obviously Jamaica, majority of our croc population is all along the south coast here. We have some small remnants up here on the north coast, but all along the west, and then we have Kingston down here in the bottom right corner. So all along most of the south coast is beautiful, kind of undisturbed habitat, mangroves, although highly fragmented. We have some nice morass swamps, free-flowing rivers, and beautiful little ponds like this one. And then when we come to Kingston, so down here in the bottom right corner, we have Kingston Harbor right here. We have about over 1 million people densely populated in this urbanized area. And in that area as well, we also have lots of crocs. So it's a bit different than our kind of swamps and morass along the kind of western and south coast. Oh, let me get the video. So we have crocs literally in this Kingston Harbor, and then they come out of the harbor, and a lot of times they use these what we call gullies and drainage systems that come through the city. And they use these as kind of little passageways. They're obviously foraging in here. And these are densely populated areas with squatting communities, houses, schools, neighborhoods, and they're constantly going under the roadways. A lot of times it's not an issue. Sometimes we get herds of people that create a scene and we have to go respond. So, let's see here. So this gives you an idea. So we have Kingston Harbor right here. This is all coastal ocean. And then in here, this is like downtown Kingston. And this is where all these drainage canals come in. So the crocs use all these little gullies to move through the city. There's various ponds and wetlands mixed in there along the coast, but there's crocs mixed in, kind of overlapping human croc habitat. So this creates obviously tons of human croc conflicts. And then in the kind of south part of Kingston, we then have our sewage ponds. This is a government facility. Give you some perspective, these are massive ponds. There's about 20 of these ponds, about a football size at least each. And they actually act as almost like a reserve for the crocs. It's like a protected area. There's a pretty good bird population there, and since it's government property, the crocs aren't disturbed. So you have the sewage ponds, kind of sandwiched between the Kingston kind of communities, and then behind the ponds is the coastal wetlands. So the crocs can easily, be in the ponds part of the year, and they can move to the coast and move up and down the coast as needed. I won't dive too much in the sewage ponds now. I'll talk about those on Saturday. So the zoo, besides the zoo, we're also the main wildlife kind of rehab facility. So a lot of times we get crocs coming to us kind of on their deathbeds. Sometimes they're already dead, sometimes they're nearly dead. So this gives us a really good opportunity to do necropsies and figure out what's going on. So I have an amazing veterinarian and my keeper vet tick team and our NEPA colleagues that are bringing these injured, sick crocs to us, or sometimes they're already dead. Like in another part of Jamaica, and I asked them to bring the body to us so we can do the necropsy there. Then we also have a ton of crocs that are found in the field, and I asked them to bring the croc to me, and they won't bring the croc to me because it's usually been baking, cooking in the sun for several days, floating in these ponds, and very well gross and decomposed. So these are just a few that we've got over the last few years. It's definitely not a pretty scene. I don't have the video for this one now. Marissa saw that one where this croc literally exploded as I was cutting it open. So just a quick idea. So in summary, this comprises 18 necropsies from 18 adults. All 15 of those are all from Kingston down here in that city area. 3 of them came from the nice, pretty South Coast area that I was showing you earlier. So what we found, so out of all 18 adults, we had 11 males, 7 females. Over 89% of all crocodiles had plastic in their stomach. 67% had some type of natural prey item. The top 3 were birds, pieces of crabs, and dogs. So that's actually not surprising. Jamaica in general, especially Kingston, there's stray dogs everywhere. And so you have crocs living in the city. Dogs are coming down those little gullies and stuff to drink water. They're literally dogs everywhere. It's actually a big problem in Jamaica. So that was a big thing. Oh, shoot, video. So this is actually one of the crocs that was from the sewage ponds. And so the frequency of occurrence is one of the more common metrics to document this because it's showing, okay, now we can say we have 89% of crocodiles. But it doesn't really cover the volume of the plastic and stuff that we're seeing. It's not just like we're finding a couple pieces of plastic here and there. Like, you'll see it's like these stomachs are majority plastic. Like, and so it's also not surprising. So we had 2 stomachs, 2 crocodiles that had no plastic in their stomach. And where were they found? Right here in this nice natural wetlands. All other 16 crocs that had stomachs loaded with garbage were in that Kingston area. And so like I was saying, you know, frequency of occurrence, that is a more common, is we can say, okay, all these crocs had plastic, but percent composition is also a more powerful tool to show how much plastic we are seeing. So for instance, this stomach in the middle, these stomachs are bursting with plastic. I barely had to cut them open, and they're spewing open. So for instance, all the stomach contents in this animal weighed 2.3 kgs. 94% of that was plastic garbage. And so it's important to include that as well. These are all different ones, forks, plastic bags popping out. So I think we need that. It's a good metric to use so we can show actually how severe this issue is. I call this guy patient 0. So this is back in 2020. Prior to that, a lot of times when our partners, the government agencies would find a dead croc, they just throw it on the side of the road, bury it, get rid of it. And so that's when I kind of reached out to him. And was partly interested in necropsies, also interested in doing like a passive diet study. So I was like, hey, when you guys find dead crocs, just bring them to the zoo. You know, we'll open them up, take a look at them here. So this guy came. He actually looked fairly healthy. It wasn't the usual dry skin, sloughing skin issues we sometimes see. But opened up his stomach, packed full of 19 plastic bags. So then that obviously created, you know, I shared that with everyone, and we're like, that created immediate priority, like any dead croc, bring it to the zoo, or if you can't bring it to the zoo, I'll come there and we'll open it up right there. Another interesting case, so this was actually just last year. Roadkill croc, or actually we're not sure if the croc was run over, and then after he was run over, the tail was cut off, or there's a good chance the croc was already dead. They had cut the tail off and then had it left in the road where it then got run over. For those who don't know, there's some people in Jamaica that believe the tail meat is an aphrodisiac. So a lot of times they'll find crocs just floating dead with the tail cut off, and then they use the tail for meat and food, or for the aphrodisiac. So, but besides the smashed head and the missing tail, fairly good body condition, healthy skin, everything else looked all right. Open her up. 21 Pampers, diapers, 3 large plastic bags. The only natural prey items right here in this box right here. 1 crab leg, another piece of a carapace, some stones and plant matter. But again, this stomach, I have a video in a sec. It was like I could barely pick it up. You know, I think it was like 6 or 7 pounds. And again, and it was just about to rupture, just coming out. And so even if we were trying to do a diet study and be flushing stomachs, we want to be able to even flush. Their gastro system's totally impacted. They're not eating, digesting anything. They can't even. Another one. This one was that actually just a couple months ago. It was got reported of a croc floating in the stream. This is like 3 days later, so we ended up pulling it at the side. It actually got killed by a hook. Someone had left a poaching, illegal hook there. We opened her up. Again, what was it? 33 plastic bags. The majority all, the feathers are right here. So this is the only natural prey items. About 200 grams of bird feathers. The rest of the nearly 1.6 kgs was all garbage. So one of the major recommendations is, this is a really neat paper. So obviously there's a lot of stuff coming up recently with like kind of micro contaminants and stuff in crocodiles, but with megaplastics and stuff, which I'll talk about here in a sec. And marine megafauna, so this is a neat review paper talking about fish, turtles, mammals, and what was the other one? Seabirds. This has obviously been a serious threat for decades now, and then over the last couple of decades, reports and publications have just been shooting up. But I think with crocodiles, since this is a somewhat new thing, it's important that we not only are documenting this, but then we're standardizing the metrics. So this is going to help a lot with obviously assessing kind of these threats, where this pollution is coming from, working with policy change, working with government officials to try to actually enact change. And so I think the standardization of metrics, and this is what the paper really stressed, is because if you look at all these marine megafauna papers, people are reporting data like in various different ways. It's made it very difficult. And so one of the things they urgently stress is the standardization of reporting the plastics across the board. So one for meta-analysis is make much easier for spatial temporal comparisons, and then obviously for policy change, mitigating these threats. And so what they're saying is obviously location, method of sampling, was it a necropsy, was it a flush sample? I was talking about earlier, frequency of occurrence is an important one. And then taking your median mass, the mass of ingested plastics ingested per crocodile. So, a lot of times we're doing other semi-content studies, you just kind of document, 6 pieces of plastic, or you got a crab, you got a snake, this, but you're not weighing each individual items. You need to weigh the individual items and, time up? Sorry. And then they say also report by debris category. So user plastics versus industrial. So user plastics, was it a plastic bag? Was it fish netting? A lot of our plastic bags possibly came from supermarkets, and that's another way you can address those threats and figure out, how can we eliminate this? Thank you.
Thank you, Joey. Can I have Miriam Boucher up here, please? All right, our next talk will be by Ms. Miriam Boucher, from South Carolina.
Awesome. I'm back. Did you miss me? I got you before lunch, now I'll get you after. So kind of on the tail of what Joey's speaking about, this is the second half of my PhD dissertation in exploring emerging contaminants. So I spoke to you before lunch about PFAS. Now I'm going to be speaking with you all about microplastics. So we have direct ways of ingestion of plastics like Joey just showed, but beyond that, our plastic waste that is produced isn't always ingested like this. It also exists in other forms because as these plastics weather and degrade in our ecosystems or are produced as primary microplastics like pharmaceutical beads or things like that, they can be ingested and accumulate in living organisms. Our understanding of the toxicological and health impacts are still kind of in their infancy. We have a hard time of fully quantifying that, but we do know that plastic ingestion can create inflammation, it can obstruct GI tracts, and it can cause other problems. In 2021, 2 professors, Dr. Stephanie Whitmire and Thomas Rainwater at Clemson University, opportunistically sampled alligator feces from golf courses and digested those samples and used simple microscopy to analyze them for microplastic. And if you look here, this is an example of 3 of the kinds of microplastics that were recorded. And so what that kind of clued us into is that there are alligator ingested microplastics, but there are no studies currently that are looking at that. So for this part of my PhD, what I wanted to look at is, could we recover and analyze alligator ingested microplastic? And can we explore alligators as sentinels for microplastic pollution? In the same way with PFAS, it's difficult to collect samples, and if you want an ecosystem level indicator, it's nice to find a compartment or a way to study it where you can kind of hit multiple levels. And in this way, we think that crocodilians are good indicators and alligators may be as well. And then again, looking at microplastic at a regional scale. So again, to remind you, we're looking at South Carolina, and what I'm going to be speaking to you about today is a subset of 33 alligators that were harvested as part of the 2023 recreational alligator harvest. So these are alligators that were hunted and were dead, and I collected the full stomach from these alligators. So I would zip tie them in the field, so in the processor, and keep them as a closed system, bring them back to the lab, and I have 12 undergraduate students that were working with me throughout this to analyze the diet for those samples. And we kept any material within the stomach that was less than 5 millimeters in size, which is the upper threshold for microplastic, down to 20 micrometers in size, which was our lower threshold for the instrument that we use. To be able to remove the organic material, we used a two-step chemical digestion process. We started with KOH, right, that breaks down fats, starts to remove some of the organic material, and we finished with hydrogen peroxide, which kind of cleaned out and got rid of the rest of the organic material. So we took bags of alligator vomit and condensed them down into 10 milliliter vials where all particles are suspended in ethanol. We used a laser direct infrared analyzer, which uses visible spectra of light to scan an aliquot placed on a slide and tell us the size, count, and composition of microplastics in the sample. So in this data set, again, we're looking at particles that are 20 micrometers to 5 millimeters in size. And we're looking at 33 alligators. I did 3 replicates per alligator, so that's 99 runs total. And we looked at almost 250,000 particles. When you look up there, it says greater than 70 HQI. What that means is that the spectra that were read from our samples had at least a 70% assurance of being positively identified against a spectral library. So on the bottom, you'll see each individual alligator's ID, and these are organized from lowest amount of synthetic materials up to the highest. And so there's a lot of variation among alligators. And so our range here in our samples was about 3% synthetic particles up to about 50%. When we remove out our natural particles, which are things like chitin or cellulose from other items that they ingest, our greatest synthetic particle across the board is rubber. Rubber is not a microplastic, it's an elastomer. And although we're only using spectral data, some of our particles do identify out as tire wear particles. To what extent, we don't know, because you need chemical analysis to be sure about the composition of exactly where that rubber is coming from. But this is a big thing, that rubber is most common in our alligator samples. When we remove rubber, we see that there are other particles present, and these are all plastics. Some ones to pay attention to are polyamide. This is nylon, most likely coming from textiles. Every time we're washing our clothing, our wastewater treatment plants are not equipped for that. We also have PTFE, or Teflon. This is also a PFAS and used in a bunch of different coatings. And then ABS, it's used in car parts and Legos. What I wanted to do is look at what might be driving this variation. So I looked at some alligator, particle, and spatial variables and ran a series of models to see whether any of these metrics predict alligator exposure. One of the big things that I thought is that coastal alligators would have higher particle counts because they have the downstream effect of plastics being moved downstream. What I did not find is a statistically significant difference. So there was no real measurable difference between the count of synthetic particles and the diversity, so the profile of particles, amongst our coastal and inland habitats. Did find somewhat of a very fine trend, not a statistically significant one, or at least a little bit, between alligator total length and synthetic particles. And so we actually saw a decreasing trend in particle counts. I think that this may be in relation to smaller alligators eating more meals frequently, larger alligators eating larger meals less frequently, and we think that diet is a primary route of exposure. When I compared this against the counties that had the most alligators sampled in the same county, there were not statistically significant differences among counties. So spatial variability of microplastic diversity indices, which was a metric of count, shape, and size, did not find that to be statistically significantly different. Another thing that we thought too was percent impervious surface, how much roadways, how much development would be there would affect what we were finding. And similarly, we did not observe any strong trends. However, it looked like there may have been the greatest variation and effect in alligators that are closer to and had lower impervious surface. Lastly, I ran this using our diet metrics, so I did individual diet items and prey groups. None of them were statistically significantly different except for one. And this is the proportion of non-target, so vegetation and rocks. And what we found is there's a statistically significant trend where there's a negative correlation between ingested particles and non-target items. So this can mean maybe one of two things. One, that if they're filling their stomach with non-diet items, they're not being exposed as much because the plastics are being transferred through diet items and not through the ambient environment. So this is one of the hypotheses I have that might be driving this. So again, unlike fish and other organisms that may be exposed through gills or membranes, crocodilians, their main exposure route to contaminants in a lot of ways is what they eat. So if they're not eating prey items, then they may not be getting exposed to microplastics in the same way. So general takeaways, it looked like our distribution across the state for our microplastic counts and diversity was kind of ubiquitous and uniform across the state. But our polymer counts and composition may be based more on finer scale, so habitat metrics, which is something I'll look at in the future, and also that polymer composition may be more driven by individual alligator behavior or trends towards what they're eating and how often they eat. As I move forward with this, I'll be looking at comparing my South Carolina alligator samples to those in other states. This is like a little teaser of just a few of the samples that we have run from different states, and even just with the small sample size, right now it looks like there could be much different profiles among states. And so looking at what does that tell us, and does it give us or correlate to information in terms of what industry is in those states or what waste management or erosion mitigation practices may be there that's keeping particles out. One of the reasons why I may have seen not statistically significant differences in this data set is I really prioritized running replicates to see how accurate our instrument was at measuring those differences. So I prioritized that. But what I did find is there was no difference in our replicates. So although I ran 3 replicates, 1 replicate or 2 replicates are adequate. And so to be able to strengthen my analyses, I'll be focusing more on bolstering the number of individuals that I run versus the amount of replicates. And so that's what I'll be doing moving forward. And so also if you're thinking about doing this or you're using a similar set of equipment, we can talk about it, but you may not need to run as many replicates as you do individuals. Again, these are all of our project partners and collaborators. Thank you.
Thank you very much. Thank you, Miriam. If I can get Anna to come up. We're going to continue on with the Clemson show with Anna Chobot.
Hello everyone, my name is Anna Chobot and I am an undergraduate student from Clemson University in the United States, and today I'm going to be telling you a little bit about my undergraduate independent research project. These are some preliminary results of where I performed a comparative analysis of alligator ingested microplastic to environmental and biological matrices. So, why is this important to study? American alligators, as we have discussed, are large, long-lived aquatic predators, and they're known to be good indicators for contaminants in food webs. So, many studies compare microplastics across diet, but there's a demand for studies that compare microplastics across more matrices. So in order to do this, I decided to compare across 4 coastal South Carolina sites, compare microplastic abundance and diversity. And I also compared microplastic count, size, and composition across 4 different matrices, so sediment, water, fish, gastrointestinal tracts, and alligator diet, to create kind of a bigger picture of what's going on and how these particles move through the ecosystem and food webs. So as for my sites, I have 4 coastal South Carolina sites. So Yawkey Wildlife Center, Santee Coastal Reserve, Nemours Wildlife Foundation, and Spring Island. Yawkey Wildlife Center and Nemours Wildlife Foundation they have less public access compared to Spring Island and Santee Coastal Reserve, which have some communities nearby as well as recreational access. So that's important to consider. But the habitat at all of these sites is comprised of historic rice impoundments and brackish marsh. So, as for sample collection, we performed gastric lavage for our alligator diet samples, as well as collecting sediment samples with the Ekman dredge and water samples. And these samples are composites of 4 replicates collected along a 100-meter transect. And then our fish samples were provided by the Strive Lab at Baruch. So here's kind of a summary table for my fish samples, including site, number of samples, mean length, mean gastrointestinal tract weight, and we have many different species included to kind of cover that variety, so freshwater species as well as saltwater species, as well as trophic variation among these species. So this is my alligator length and diet weight summary table. Just to kind of keep in mind my sample numbers here, if this is a smaller data set, we still need to bolster sample sizes for Spring Island. So as we get more results in and run more samples, this data set will become a little bit stronger. So as for sample preparation, it's very similar to Miriam's study, a lot of the same methodology. The only true significant difference is that my water and sediment samples were only had a 1-step chemical digestion, as opposed to my alligator diet samples and my fish samples, which had a 2-step chemical digestion. But very similar process with sieving and then chemical digestion to get the organic material out of the samples and get them prepared for LDIR analysis. Okay, so for particle type, this is my, this is my synthetic total particle counts. So 7,525 total particles were analyzed across my samples, and 66,000 of these had a 70% or higher identification quality. And there's some significant findings within this to note. So with my sediment samples, they have significantly higher number of particles per sample, as you can see, and as well as a higher mean diameter and median diameter. So that's good to take note of. And as for particle type and proportion, so fish, sediment, and water samples all had kind of a similar distribution of synthetic particles compared to natural particles. However, my alligator samples showed a surprisingly lower synthetic particle count, and this could be due to stomach flushing, which is taking a snapshot of the stomach contents at that particular time, as well as it's not including tissue, and my fish gastrointestinal tract samples included tissue. So that could be a driver of this difference here. Additionally, alligators on the South Carolina coast primarily consume crab and shrimp, and so that additional chitin could be causing this skew. So now we can look at the composition by matrix. So we have my fish, gator, sediment, water, including the rubber particles. So as you can see, similar to Miriam's results, the rubber kind of blows everything out of proportion. So we removed that rubber to kind of take a look a little closer at the plastic particles. So lots of polyamide, similarly to Miriam's study from flows, and then also PTFE, Teflon, polypropylene, and a significant amount of magnesium stearate in both the fish and the alligator samples, which is a synthetic salt used in plastic production. So why that is accumulating in the fish and alligator samples is something to look into. Additionally, it's important to note that those fish and alligator microplastics have a very similar polymer composition. So this could be important to consider. So as for my polymer composition, I ran an MDS, and the main takeaway here is that there is not much overlap between my sediment and my alligator samples. So that's the true difference there. But my fish and my alligator samples have a lot of similarity. So what this can indicate is an important food web dynamic that supports that alligators are good bioindicators, as the microplastics found in these fish samples are very similar to the microplastics found in my alligator samples. Okay. So conclusions and future directions. There was no significant variation in particle count across sites. There are similar microplastic abundance and diversity across sites, but differences across matrices. And alligator and fish samples show very similar polymer composition. And there's a significant difference in polymer composition between alligator diet samples and sediment. And additionally, fewer synthetic particles in alligator samples, which again could be due to methodology. Thank you very much. Big thank you to everyone who supported me throughout this project, especially Miriam and Dorothy, who helped a lot with my field sample collection.
All right, thank you, Anna. I apologize for missing to announce this, but for those of you that are following the student presentations for scoring purposes, Miriam is a student, Anna is a student, as is our next presenter, Dorothy Aldridge, who will be carrying on the Clemson show.
Hi everyone, my name is Dorothy Aldridge and I'm a master's student at Clemson University. And today I'm going to talk to you about comparing PFAS recovery and analysis from alligator blood among collection supplies and a biological alternative. And so the lead author on this paper, Umnia Bubad, couldn't make it here today at the CSG, but she's actually born and raised here in Morocco, and she conducted this research last summer at the University of Florida alongside Miriam Boucher and myself and the rest of our team. So I'll give you a little background on the contaminant that I'm going to be talking about today. If you went to Miriam's presentation this morning, then you already have a little bit of an idea of PFAS. But PFAS are per and polyfluoroalkyl substances, and they're a class of over 5,000 different synthetic compounds characterized by this long carbon chain bound to fluorine atoms. And this carbon-fluorine bond is really, really strong, and it allows these chemicals to stay persistent and stable in extreme environmental conditions, which is why they're often called forever chemicals. And PFAS partition to proteins that are found in the serum and plasma of blood, and so this affinity makes blood sampling for PFAS an appropriate method. So, blood collection for PFAS is conventionally done using blood collection tubes or vacutainers, and these contain different additives that prevent or facilitate clotting and separate out the plasma and the serum from whole blood. And this creates a more simplified matrix that can help us during PFAS extraction and analysis by separating our analyte from interfering substances. And kind of the common vacutainer tube additive is lithium heparin. So another method for looking at PFAS in blood is microsamplers. So the two I'm going to talk about today are dried blood spot cards and Mitra samplers. So dried blood spot cards are cotton cellulose cards with 5 circles that indicate where to place blood aliquots. And then Mitra samplers are a type of volumetric absorptive microsampler that has a hydrophilic polymer tip that you can dip into whole blood, and that will absorb a fixed amount of blood. And both of these samplers use less than 0.5 milliliter of whole blood, and they can be stored dried. Alternatively, we looked at biosampling sangivorous or blood-feeding external parasites as a proxy for whole blood. So this has been done before in a study on ticks as sentinels of environmental PFAS, and these ticks had multiple host species throughout their life at different trophic levels. So looking at the ticks kind of gave an idea of the general contamination present in that ecosystem. And today I'm going to talk to you about leeches, because leeches are a common parasite found on crocodilians. So our objective is using American alligator blood to validate recovery and profile of PFAS among conventional and emerging blood collection supplies. And so for this, we use 5 different vacutainer tubes: EDTA, which is whole blood; lithium heparin, which is a type of plasma separator; sodium citrate, another plasma separator; and the plasma preparation tube, as well as serum separation tube, and as well as our 2 microsamplers and leeches. So our study area was the United States South Coastal Plain, and we sampled 21 different alligators across 6 different sampling sites. And this was in North Carolina, South Carolina, Georgia, and Florida. So we collected 5 to 30 milliliters of whole blood from the post-occipital sinus, depending on the size of the alligator, and we distributed that equally into 5 different vacutainers. And then we transferred 1 milliliter of blood into a vial for our microsamplers. And so vacutainers were kept on ice, and plasma and serum were centrifuged to separate the supernatant into our samples for analysis. For our microsamplers, we pipetted 40 microliters of whole blood onto 3 of the 5 blood spot circles, and we took the weight before and after. And then for the Mitra sampler, we just dipped the tip of each of the 4 Mitras into the whole blood vial, and we also recorded the weight before and after. And both of these samplers were stored dried at room temperature for up to 3 months before PFAS extraction. And then we inspected the alligator for leeches. So leeches are often found on more delicate parts of the alligator, so under the jaw or in between fingers, sometimes in auxiliary regions or on the hind legs. And we tended to find juvenile leeches in the mouths of these alligators, as you can see here. We found 3 different species of Crocodile leeches, which are primarily ectoparasitic on reptiles and amphibians. And so this leech we named Leechzilla. It was a huge leech that was found by Anna under a partially attached osteoderm with her brood, or their brood, because leeches in Crocodile are hermaphroditic. So our leeches were homogenized into these leech smoothies, as you can see on the left, and then we cut out the blood spot cards and we popped off the tips of the Mitra sampler into vials for PFAS extraction, and this was done using paired ion exchange, as well as protein precipitation, which isolates our analyte, which is PFAS, from the surrounding interfering matrix. And then we ran our analysis using liquid chromatography, tandem mass spectrometry, to quantitate the response peaks of 30 different PFAS compounds. So what we found is comparable PFAS concentrations amongst different blood, vacutainer blood tubes, as seen on the figure to the left. And so what this is showing is the top 8 most abundant PFAS, and these accounted for 95% of the total PFAS concentrations in our data. And we found highest recovery concentrations in the serum and heparin vacutainers. And you can see there's one PFAS FBSA that we found solely in whole blood, And this is a short chain PFAS, so it could be due to a higher affinity to red blood cells or just a lower affinity to the proteins found in plasma and serum. We also looked at total PFAS equivalence values in comparison to lithium heparin as our reference. And so for that figure on the right, the red line shows the point at which the total PFAS in the different vacutainers equal the total PFAS in lithium heparin. So you can see we saw very similar equivalence between plasma, serum, and whole blood, with whole blood having a larger dispersion and citrate performing lower, although having less dispersion and higher accuracy between samples. So then we also looked at our different microsamplers relative to concentrations of whole blood, and we found lower recovery in both Mitra and blood spot cards, with Mitra performing better than blood spot cards. And we also found 2 additional non-detects in our blood spot cards, but these were associated with alligators that had overall lowest concentrations of PFAS in our data set. So then we ran an NMDS to look at how sample type affects the composition between in all of our different samples. And we found, so here each of the points are different samples, and they're symbolized by sample type. And we found overall high clustering, which is good. It means that we're seeing the same PFAS composition across our different sampling types. And sample type described about 12% of the variation in our data set. As you can see, leeches had highest in-group variation, which is the blue dots. They have like a wider dispersion on our plot, and they're most highly associated with whole blood, which is the red dots. And we also saw that Mitra and blood spot were most highly clustered in tight clustering, and so this indicates that our microsamplers have overall most similar composition. We also ran a linear regression to understand if leach size had an influence on total PFAS concentration, and we found a weak negative association with leach length. And as you can see, I pulled up the picture of all of our leach smoothies. They're very different colors. So this might have something to do with the amount of blood meal that was present in our leaches at the point of extraction. Okay, to conclude, we found highly comparable blood profiles, PFAS profiles across our vacutainers, with lithium, heparin, and serum performing the best. Our microsamplers had lower recovery concentrations, but high profile agreement with whole blood. And our Mitra samplers seem to do better than our dry blood spot cards. Now this is particularly important and advantageous for our field because microsamplers can be stored dried, so they can be helpful in remote locations where we might not have access to refrigeration or laboratory freezers. And they also only require very small quantities of blood. So we can do minimally invasive sampling with a pinprick or maybe in association with scoop clipping, as you can see on the picture to the left. So for leeches, we found representative profiles which suggest that leeches could be a viable biosampling alternative, and they were relatively independent of size class, so you could sample juvenile leeches or adults. That being said, there is limitations with life history of the leeches. Placabdella species are generalists, so they could have multiple host species throughout their life, and they can live independently from their host species, so we may be getting more environmental influence. And I'd like to thank Umnia and shout out to Miriam for all her hard work in this, and thank our collaborators and vendors.
Thank you, Dorothy. And our final speaker of the session is Marisol Buenfil Rojas, who is joining us from Japan. And she will be Pretty sure she's in Japan. Said she's joining us from Japan. All right. Excellent.
Yeah, I'm joining from Japan. Okay, can you hear me? Excellent. Okay. Well, good evening from Japan, and well, good afternoon there in Morocco, and if you're watching, Mom, good morning from Mexico, and well, to my biggest fan. And thank you for the opportunity for this presentation today. And well, my name is Marisol Benfil Rojas, and today I will show you how we can use discrete transcriptomics to identify molecular biomarkers of environmental stress in wild crocodiles, and more importantly, how this can become a practical tool for conservation and field monitoring. So in ecotoxicology, we are very good at measuring contaminants, as well as you witnessed today with many of the research that our colleagues have presented. But we are still not very good at measuring biological effects. We are kind of having a critical gap here, because exposure alone does not tell us what is actually happening to the organism. So the question becomes, can we measure effects directly in wild populations? One approach to address this is landscape transcriptomics. This framework uses gene expression from wild populations to connect environmental variation, molecular function, and conservation outcomes. So what predicts is that we should find a shared core response in our individuals of study. And then if we have site-specific variations, they will be driven by these local conditions. So based on this framework, we have this question, why transcriptomics? The advantage of using transcriptomics is that instead of looking at one biomarker at a time, we capture thousands of genes simultaneously. So rather than seeing one piece of the puzzle, we can reconstruct the entire biological response. But then there is here an important challenge, as you can see from today's presentations, the sampling part. Because most of the times when we do this kind of effective studies, we use blood, because of course it's by excellence the first compartment to use for this kind of studies, or we use internal tissues. But of course, if we want to conduct non-lethal tissues, this becomes a challenge. And in crocodiles in particular, caudal scutes are often considered structural or inert tissues. So this raises an interesting question. What if we can actually use scutes to measure biological responses to contamination and not only for exposure? So to test this, we designed a study linking environmental exposure to molecular responses. What we did basically is we sampled wild populations in Mexico, more or less crocodiles, wild populations, along with captive individuals as reference. And by using non-lethal scutes biopsies, we generated RNA-seq libraries from these scutes. And then we applied differential expression, pathway analysis, and network analysis. So using this framework to first measure the effects, now we can ask, is there a consistent biological signal across these populations? And well, long story short, since I don't have much time, yes, we can. First, what we can see here in this slide is that we identified over 18,000 expressed genes with around 4,000 differentially expressed genes. The important part here from this slide is that the PCA is showing that there is a clear separation between the captive and the wild individuals. And most importantly, what it's telling us that this is not noise. This is a structured biological variation that is actually consistent with the landscape transcriptomics framework, because we are finding something very interesting, which is that we actually have a conserved core response about 85 genes. And then we also have a large set of genes that is corresponding to this site-specific variation. So altogether, what this is suggesting is that environmental context does not create completely different biology. So we're not going to have like perfect clusters for our wild populations, but rather we will have modulated shared biological programs. Of course, to understand how these responses are operating at the individual level, and since we are approaching something in ecotoxicology called adverse outcome pathway, which is basically we're measuring the effects first, and then we are trying to connect this to the contaminants. So what we did now is that we summarized this gene expression into the biological pathways, but instead of using the classic approaches, we used the single sample gene set enrichment analysis. And then in simple terms, what we did is that instead of looking at the single genes and then trying to connect the pathways, we are evaluating an entire biological process within each individual. What we found is that there is a high variability, and this is not random. For example, what you can observe here in these slides, you have oxidative stress process and then detoxification pathways and immune inflammatory responses. But what arises across this is that this process is particularly for oxidative stress and xenobiotic and detoxification. There is a variation that is site-dependent and on environmental pressures. And importantly, something that arises that is very, very interesting, and everybody was asking me, but in the part of oxidative stress, your captive individuals are having a lot of oxidative stress. And no, this is a wrong interpretation. What we are actually observing is like the baseline of the levels on the oxidative stress genes that we should have from the captive individuals. And then from the two populations that are different, we are observing suppressed oxidative stress responses, which is actually reflecting chronic exposure to the contaminants. So to move beyond these individual pathways, because first we have something now that this is not random, and this is showing, of course, relationship to oxidative stress and xenobiotics, but to know how these genes move, we applied co-expression network analysis. This allows us to identify a group of genes rather than, again, single genes, and how they act together as coordinated systems. And of course, we made the contrast between captive and wild status. So what these modules are showing is that we have a strong biological coherence, and particularly that in wild individuals, processes like extracellular matrix remodeling and lipid metabolism are the ones that are dominated by these groups. So now we are capturing system level regulation. And finally, to put this all together, to get this biomarker panel associated to SCUTs, we built a network-informed biomarker panel by using machine learning approach. And we used the module membership information, differential expression, and individual level discrimination, which is basically the analysis that I showed you. And up to this point, what we identified clearly is a consistent biological signal, which is translated into 10 candidate genes that are actually related to oxidative stress and also to xenobiotics and detoxification process that perfectly separates captive and wild individuals. And these genes are not genes that are classically studied in other studies that we have observed for this kind of effect approach. So the next question is, what is driving this? So what we did is that right now what we are doing is that we are integrating the transcriptomics part with the metabolomics part. And what we are observing, even when we have different factors such as sex and such as, for example, captive wild populations, we are observing a consistent pathway level patterns across both layers. What this is telling us is that signals are biologically coherent. And again, this is not a statistical artifact. We are actually having consistent signals. And finally, this is where the things become particularly powerful. Because now that we identified these pathways, which we call in the adverse outcome pathway, we call them key events. Now that we identified the key events, we are able to integrate contaminants, pathways, and metabolites together, and even hormones, to finally get structured associations between exposure and biological response. In other words, we move from simple correlation to interpretable exposure-response relationships. And finally, we are applying a machine learning model prioritization that is actually reducing this complexity for all this information, thousands and thousands of genes, hundreds of metabolites, and of course the contaminant information. And then we identify a small number of highly informative biomarkers. So these biomarkers retain the traceability to contaminant exposure. For example, what I'm showing here, traceability to the POPs, resulting in a practical and interpretable biomarker panel. So what does this mean beyond this data set? It means that skewed transcriptomics is becoming a working framework for ecotoxicological biomonitoring. And we're not only detecting transcriptomic differences, we are building a scalable way to connect non-lethal sampling, molecular responses, contaminants, and future multi-species applications. So this is still being refined, of course, with expanded datasets, because I just received new data for metals and POPs. But the core framework is already working. And this is the practical message I want to leave with you today. So you don't need to build any of this from scratch. So please consult the QR codes if you're interested, because we already have a non-lethal RNA extraction protocol from Scutes published. We have a reproducible RNA workflow that I have deposited in GitHub, so you can consult that. And currently we have in review a validated transcriptomic signature, which is part of what I showed you today. And then we are working on the exposure-linked biomarker panels and ongoing AI and adverse outcome-based modelings. So what I need now from you, from everybody, not just you in Morocco, but everybody in the live stream, I need field collaboration. So I need access to sampling sites, crocodilian populations, and people interested in applying this framework to other systems, because now that we validated this in Morlet's crocodile, the important part for me is to try to apply this to other crocodilian species and other threatened reptile species. So this is my invitation. If you have populations, questions, or conservation contexts where this could be useful, let's talk. So thank you very much for having me today. In the QR code, you have my email.
Thank you, Marisol. It was a great presentation. If I can invite all of the session speakers up on stage, we have time for a very short panel. And as they're doing that, I would like to thank them all for their diligence in their talk timing and working very hard to get us more or less back on schedule. All right. Does anybody in the audience have any questions? Is there a microphone over here? There's not a microphone. Okay. There is? Okay. Oh yeah, just right there. Thank you. All right. So I have a question to get us kicked off. I'm just curious what, especially for the people whose lab they're doing work on both plastics and other non-plastic, or let's say non-physical contaminants, so chemical contaminants versus physical contaminants. Which do we think poses a greater threat to crocodilians moving forward, considering the tenuous state of the environment around the world today?
I don't think you can ever even say that, because all we're doing is taking a snapshot of a cocktail of things that our study species are exposed to at any given point. Like, I think one thing that exists in other aquatic study species is like multi-contaminant data sets, and we need to have more of that and trying to pull them together. But I don't think there's a real answer to that, because we're only quantifying a very minute amount of what exists there, and that doesn't take into account other things like, you know, oxidative things. These are just contaminants. So that's my 2 cents.
I also want to add that they're not mutually exclusive, so different organic contaminants can absorb onto microplastic particles. So microplastic pollution and contamination in these crocodilians may also include a lot of these other contaminants.
Okay, merci. My name is Ilasa Wedrago. I want to know in what condition a crocodile eat a plastic in the reserve. It is a lack of food or I don't know.
Yeah, I think in that area, it almost seems like the crocs are starving and they're just consuming anything they can that has, from what we've seen, is a lot of it's been those plastic bags or some type of human waste that has probably a good or somewhat tasty odor or something on it, and they're just consuming anything because there's, like you said, there's not a lot of mammals there. There's a good bird population near the waterways, but in those Kingston gullies, they don't seem to be finding the food that they need. I think they're just consuming anything they can. And because we saw very little actual prey items in those stomachs, and although we found numerous dead crocodiles that are obviously illegally killed, we had several crocodiles that were randomly found dead of unknown causes. In the necropsies, there's probably at least 3 or 4 after necropsies. I'm almost for certain they died from impaction and even gastric rupture from the plastic.
Hello? Okay. Hi, Marisol. This is a question for you. It's Helen, by the way. You did amazing.
Okay.
For your RNA extractions, for those who don't know here, RNA tends to degrade really, really quickly. So when you got your RNA extractions or your RNA samples, did you get your tissue samples and then do your extractions in the field and bring those into the lab where you were doing it, like all of the laboratory analyses and the laboratory processing? Or is it something where you had the actual tissue samples, stored it, brought it with you into your lab in Japan or wherever you did your actual extractions? Because if you're looking for collaborators to send over samples, I would assume that if you're going to get it straight from the tissues, then there are some logistical challenges in terms of permitting to bring through multiple borders across countries, versus if you were able to do all of the extractions and everything inside on country. And if that's possible, what is the best ways for those who want to send samples to you to preserve? Because RNA later is very expensive.
Yeah, thank you very much for the question. And actually, it was very challenging at first to set up this because, particularly in Mexico, of course, we didn't even have access to dry ice. So what we have to do is to use RNA later, which is something that I optimized here in Japan, that we work with RNA later and we use portable freezer. But at first we were using only ice, like ice and the ice box. So one thing is that I know perfectly the challenges that we have in the field in that sense, and also that part of what you're mentioning about bringing the samples. Yes, of course, it was a huge challenge to bring the samples here in Japan because of the constraints, of course. So I think that depending on the CITES appendix, because Crocodilus motileti is in the appendix too, which was not really hard for us to bring here to Japan. But if we're talking about another threatened species such as Gharial, for example, I think it's better to process in site. So that part of the collaboration might be challenging, but at the same time, I do have experience on how to manage these samples and the part of the exportation. So again, if you're interested, we can talk about that, because after my 3-year excruciating experience to bring the samples here, I can tell you how to do it.
Sorry, Matt. Can I add something? Okay, so we work with blood samples, and RNA later is not good for that. But we can, transport samples in the middle of the extraction to United States, and then finish the protocol there, and it works perfectly. Of course, you need a liquid nitrogen tank in the field, but it's not hard. I mean, you can take a small one, and we do it. So, but that is a different sample that the one that Marisol works. So it's for different things, maybe, but there are more.
By rubber to tire wear particles as a point source, unless there's a hit quality of 95% and up. So we do have particles that are representative of that. It is likely that tire wear is one of the greatest contributors to those rubber particles, but we would need a chemical analysis to really say with surety that that's the case. The other thing to consider for rubber is, and this is an emerging contaminant on its own, there's an additive in rubber called 6-PPDQ. Right now in North America and the United States, that's gaining attention because there's been a lot of related fish mortalities in relation to exposure to 6-PPDQ. So this is also something that might be a future pathway, would be to link our rubber ingestion metrics with measuring 6-PPDQ if it is accumulating in alligators as well. But that is another contaminant. But is that an adequate answer? Okay.
Yeah, my question is kind of for the entire panel. It's kind of a generic question. All this work that y'all are doing are absolutely fantastic, but given the results of this work, how do we apply this? What do we do next with it? How do we invoke change in policy? What are your thoughts on that?
I mean, my plan is to get this published as soon as possible with more of a focus on Jamaica, obviously. There's a few years ago, they actually banned what they call scandal bags, which are like the plastic shopping bags, because of issues like this, but still common folk and everything still use the small plastic bags for putting items into the store. You go to the supermarket, you put your vegetables in these little plastic bags, get home, those get thrown away immediately. So I think if we can get this paper put out, get enough attention to it, we can enact some policy change in Jamaica to get even more of that stuff banned and eliminated, because that was one of the #1 waste items we found in our animals.
I think one thing, I guess one of the strengths for alligators is there's commercial and recreational harvest across the U.S. So I think there'd be some finessing to not create a scandal around reporting things like PFAS or microplastics in alligators from a consumer threat front. I think that would have to be finessed. But being able to draw that kind of direct link to people are eating these, people are wanting to hunt them. In most states with recreational hunter harvest, the demand for tags has only increased over years, and there's more interest in it. And so I think we could leverage the interest and the sustainable use aspect, at least in the case of alligators, to kind of drive more public consciousness around potential transfer risk, not in a crazy way oh, this is a huge health scare, but just as an analog between they're picking this up, you could pick it up from them, but also they're still eating all the same things you are in those systems. And I think that's one way that we could leverage some of this data, at least in the case for alligators, to push for better public policy around some of this.
Well, it's quite challenging because in toxicology we are always running behind everything, because the amount of chemicals and physical agents in the environment is really huge. And we are, as Miriam said, evaluating one or just small mixtures, maybe of chemicals, but you know that, for example, microplastic are used by chemical as vehicles, so that is a combination between physical and chemical agents acting on animals, and that is very, very difficult to evaluate. But for example, in our case, the results we get for some pesticides, for example, for glyphosate, was used for the International Agency of Cancer to reclassify the glyphosate as a possible cancerogen. So I don't know. I know, and I think that we can make small things that in the future can have a real impact.
Excellent. We'll just have time for one more question.
Just can't help getting out of my mind that the man who invented plastic bags is the crocodiles did not seem to be thin or emaciated and with a full stomach. That is a question. Were they thin because they obviously their stomach's full of plastic, but when they eat more plastic they must be getting some nutrition or something? That was one question. And the second one, have you ever seen any sign of the crocodiles suddenly starting to try to regurgitate? Heard of that happening with some things, with fur balls in the stomach. Is suddenly the croc starts, regurgitates the fur balls. Has there been any indication of that happening with these full stomachs full of plastic?
I'll answer that one first. We haven't had any regurgitation because of the plastic and stuff, not that we've seen so far. We had one that was captured illegally by some people, and the authorities weren't able to get there quick enough, and I think they had it restrained, not properly and everything, and out in the sun, and over time, by the time it got to us, it had regurgitated on transport. But the interesting thing, it had some septic issues, something going on, because it was all green slime and stuff coming up, from the system. But the interesting thing with that was that that was one stomach that had no plastic in it, actually. So we're not sure what their bottom line is. A lot of the ones I showed on there, we did have several crocodiles that were emaciated that I wasn't able to show there, that were full of plastic as well. So I don't know if those were maybe ones that had plastic in their stomach longer, and some of those ones were more recent, but it seemed like it accumulated quite a bit, because I had several that not only emaciated, that were freshly dead, and their skin was sloughing off, all their bones sticking out, various other issues. So definitely seemed like some whether contamination and impaction, but it's interesting with some of the ones that were bursting out with plastic that they were not skinny. So I don't know if those are recent or what, but it doesn't seem like, I don't see how their whole system was totally impacted, like how anything could be passing through there. Thank you.
All right, thank you, everybody. That was a fantastic session. There is a coffee break. Please try to come back in 10 minutes, all right? 10 minutes. Thank you, Marisol.
Thank you so much. So I think I gave you back the control, so just confirm.
Yes, you did.
Okay, perfect. So thank you very much, Matt, and thank you very much for responding all my emails through thick and thin, and I'm really sorry that I couldn't make it, unfortunately, for the situation. Yeah, I was happy to do so. Thank you.
Bye. Yes.
I wasn't able to upload my presentation because I have a lot of videos, so yeah, I have it tomorrow, but I wanted to check
I'll take it now.
later.
Don't want to...
I think we need a new battery. Can I help you?
Yes, we need to get the audio. I've got videos in mind.
You sure it's not here already?
No, because I've got videos. I uploaded one and I was told to bring my own, so I'm going to connect through my own laptop.
I don't think we can do that. Go back to what you give me your thing and I can
No, I don't know. Can you do that? Yeah, sure.
Yeah,
Yeah, no,
just put it
no.
on that. I'll put it on this.
Can you do it? I'm so nervous. I'm not like, like seriously.
Exit out of this. All right, so you have 31 slides with video.
Trust me, it's fine.
Okay. This is the next session. And just save as to the thumb drive. Save a copy is fine. And then you have to find the thumb drive.
Where? Browse.
Your computer. And you can also do it up here maybe. USB, yeah. Yeah, you can save. Jeffrey, who have you been talking to about this?
Well, over email, because...
If I need to know who. Was it like the conference organizers, the
Yeah,
CSP translation?
So I uploaded it, but you said you couldn't upload videos, and you had to bring your own laptop.
working on this stuff, people use these terms in way too many ways, or lots of ways. So have a look at how we've thought about it. It's there to react against or think with, but please have a look at those definitions. So without further ado, I need to find our first speaker. So first up, we have speakers from Australia, India, Indonesia, Papua New Guinea, unfortunately Zambia and Nepal no longer. So first we have Kirstiana Ward, who's managing lawyer for the Environmental Defenders Office's National Early Legal Intervention Team. Christiana contributes to croc conservation in Australia, contributes to parliamentary inquiries, coexistence strategies, yay, coexistence, and community-led law reforms. So take it away. So that, wait a minute. You should be on there, hang on. I need to select you there. Sorry. No, they're just not there. I need to get rid of me. Yeah. Is that okay? Yeah.
Gurunbunan, G'day. In the languages of the Gimoy, Wallabara, Yidinji, and the Irukandji people, I acknowledge them as the traditional custodians on the land where I live and work, in Cairns, far north Queensland, Australia. And I pay my respect to elders past and present, and to our Moroccan hosts. I'm an environmental and human rights lawyer with the Environmental Defenders Office, specialising in coexistence, cultural rights and legal frameworks for human-wildlife conflict. I'd like to take this opportunity to acknowledge Amanda French from the Community Representation of Crocodiles for her contribution to this presentation, and to David White for his crocodile images throughout. The EDO is a community legal centre working across Australia and the Pacific. We use the law to protect people, nature and climate, and hold decision makers to account. Our vision is a world where nature thrives. This presentation is a story of saltwater crocodiles, community and culture, embedding first laws and human rights into the Queensland environmental laws to protect both crocodiles and people. Saltwater crocodiles inhabit much of Queensland's populated coastal communities and the far north Cape of Queensland and Australia. Saltwater crocodiles are protected in Queensland and remain vulnerable after being hunted to almost extinction by the 1970s. When a human and crocodile interaction occurs, the Queensland Government has the powers to determine a crocodile as a problem crocodile, and those protected animals are mostly removed from the wild and euthanised. As more people live, work and play in crocodile habitat, we were seeing an increasing number of crocodile and human interactions that were reckless and avoidable, with crocodiles paying the price for these behaviours. This next video is confronting.
Camera rolling as he edges into the shallows of the...
I'll just let that go a bit quieter, thanks. Yeah, right down. Now we need to go back. Sorry, everyone. But now I need my presentation slide back on. Sorry. I did try and...
You just click through
yeah. No. Right.
Camera rolling as he edges into the shallows of the Bloomfield River, Alistair McPhee almost films his own demise. A crocodile suddenly grabs his leg, then lets go and quickly snatches his dog. Alistair punches the reptile, but the croc slips away. The 37-year-old staggers back to shore.
In February 2023, this man ignored crocodile warning signs, local ranger advice that there was a 5-metre crocodile in that location in a First Nations community. He filmed himself, a pet dog was killed, and 2 vulnerable crocodiles were later destroyed under our problem crocodile laws. We learnt these 2 crocodiles were culturally significant to the Kuku-Yalanji people. Their removal caused deep harm to their community, culture and country, and required a whole of community response. The Bulgamu is the spirit of the Kuku-Yalanji people. This harm to their culture demonstrated that cultural rights and first laws must be embedded into environmental law and our crocodile management frameworks. Following the removal of that dominant male, the aunties described ecological destabilisation and increased risk around customary food gathering practices. This meant they couldn't collect their mussels, oysters and fish, use their supermarket, their waterways, until a new dominant male established territory. This issue was not simply just crocodile removal for public safety, it was one of culture and ecological consequence that followed. Following the incident, hundreds of community members wrote to government calling for accountability. The government response was Alistair McPhee had not breached any laws for the death of his dog Molly or the 2 crocodiles. Fear-driven media narratives were shaping crocodile policy and public perception, both locally and globally. In response, Croc Queensland was formed to promote evidence-based coexistence and began working with the EDO to explore legal reform pathways. Bob Irwin Sr., Croc Queensland, EDO and traditional owners united to take the issues to Parliament, advocating for law reform to close critical legal gaps in crocodile management. We identified major legal inequities. The framework focused on crocodile behaviour rather than the human behaviours driving the risk. Enforcement was weak, attractants poorly regulated, and cultural rights largely absent from the decision making. In Queensland, our Human Rights Act protects the cultural rights of our First Nations people and therefore applies to culturally significant crocodile management decisions. This means cultural rights must be properly considered in problem crocodile decision assessments, and traditional knowledge must lead management actions. It is unlawful for the Queensland Government to not consider cultural impacts when considering what to do with a problem crocodile. So we travelled across First Nations communities in crocodile habitat to listen, learn and better understand coexistence on country. These communities had all experienced problem crocodile removals. We facilitated conversations centred on cultural impacts, coexistence and how First Nations communities have lived safely alongside crocodiles for millennia. This outreach reinforced that communities who have lived with crocodiles for over 60,000 years hold a significant knowledge around coexistence and safety. We heard consistent concerns about limited consultation and the need for authentic engagement across the crocodile management framework before a crocodile is removed from the wild, not after. We took the findings to government again to advocate for stronger policy and legal reform, recognising that without addressing these gaps, another serious incident was inevitable. Shortly after, a visiting doctor was killed near Cooktown, far north Queensland, following ongoing human attractants and feeding behaviours for photo opportunities. The culturally significant crocodile was later destroyed, and the tragedy became the catalyst for the government to strengthen crocodile laws in Queensland. In September 2024, the Queensland Government introduced new crocodile-related offences, significantly increased penalties and expanded powers for conservation officers to move people away from crocodiles. The reforms marked a shift from managing crocodile behaviour to managing human behaviour. The reforms targeted feeding, habituation and unsafe interactions in crocodile habitat while strengthening enforcement powers. While significant progress was being made, strong political and community opposition remained. During this period, a fifth crocodile culling bill was introduced in Queensland, calling for an expanded culling and removal powers in the name of public safety. In response, an unprecedented coalition of First Nations leaders, scientists, conservation organisations, crocodile industry representatives and advocates united in opposition. For the first time, these sectors spoke with a largely unified voice, and much of the evidence presented was accepted in the committee's report. The bill was rejected, recognising the long-term safety requires coexistence, education and shared responsibility, reinforcing a message we have long championed. Queensland does not have a crocodile problem, it has a people problem. The committee acknowledged broad-scale lethal management was not the solution. Recommendations focused on stronger public education, improved risk management, authentic engagement with First Nations traditional owners, and evidence-based management grounded in science and community knowledge. Despite recent reforms, significant challenges remain. Our current case involves a culturally significant crocodile made famous by Steve Irwin, known as Old Faithful. Old Faithful was from Rinnu National Park, an area jointly managed with traditional owners and home to Queensland's most important crocodile habitat. Increasing risky behaviour by visitors again raised tensions around crocodile management, continuing to challenge the balancing of public safety, conservation and cultural rights within legal management frameworks. This case highlights the failures of our government to act appropriately under our laws to engage with traditional owners and explore management actions that this now is a human rights problem, but we have not just a crocodile issue.
Some context on the crocodile. Nicknamed Old Faithful, the 4.5-metre reptile is revered by traditional owners across Cape York.
You can't hear this. This is a story about Old Faithful. He lived in his waterhole. People were behaving poorly. He was over 80 years of age, very culturally significant to the Kookawarra people and the traditional owners who manage this national park. The park is actually owned by them as Aboriginal freehold land. The government made a problem crocodile assessment. They, we believe, and hold serious concerns that they collected the wrong crocodile and removed him from the wild and transported him to Cairns. They didn't engage with traditional owners other than within emails over 2 hours and said it had to be done for public safety. Owen here is a beautiful man. He said, yes, you can remove the crocodile because he believed it was in imminent risk. He didn't know that it was Old Faithful that they were taking. There is no single solution to human-crocodile conflict. Effective management must reflect the realities of each landscape, community and cultural context. The most successful approaches are holistic, balancing our laws, conservation, science, cultural rights, public education and shared responsibility in crocodile habitat. Throughout this work, I've witnessed a powerful shift from fear and misinformation towards greater understanding, respect and coexistence. The strongest outcomes emerge when laws, culture, science and community voices work together. And we believe that global human rights frameworks may provide one of our strongest pathways forward to protect crocodilians and people in an anthropized world. Thank you.
Super, thanks. That was some time. And could we have Yashendu? Going to find you. Eek. Huh? Can't get rid of this. Try that again. Yeah. Where's the mouse? There's no mouse, is there? No control escape? Got it. Okay. There's the box. Okay, I'll briefly introduce Yashendu while he does the tech. He's way ahead of me. So Yashendu Chinmay Joshi is a PhD student at Muséum National d'Histoire Naturelle, sorry French people, in Paris, France. His research explores contemporary and historical interactions between crocs and human communities in Gujarat, and yeah, really looking forward to this.
Thank you very much, Simon. Thank you all for attending my talk. I'm a second-year PhD student at the Nationalist Museum in France, and today I'll be speaking about the work I have done with understanding crocodile behavior and their interactions and relationships with human communities in central Gujarat. Interactions between humans and crocodiles are increasingly becoming very common, particularly in a country like India with over 1.4 billion people. And moreover, the case of marsh crocodile is more interesting because they are known to inhabit a wide range of habitats. So apart from the national parks and wildlife sanctuaries, majority of their population is present in anthropogenic environments such as villages, cities, towns, where they live in canals, ponds, rivers, tributaries, and reservoirs and dams. And in fact, in the part of India where I work, in Western India, in the state of Gujarat, they are known to use extensive irrigation canal network to move from one place to another across the state. So basically, they are using these irrigation canals as their highways. Another thing that creates an important aspect here with crocodiles is the cultural integration. So from ancient Egyptian to Mayan civilization to the contemporary Indian cultures, crocodiles have long been a very important part of human cultures and civilizations. However, most of the studies focused in the region where I work are looking more into negative interactions and conflict, which is not without an empirical basis. However, they offer only a partial account into human-crocodile relationships. Therefore, in recent years, in multi-species anthropology, they have coined the term more than human life worlds, where these are sites of relationality, learning, and co-production of knowledge between humans and non-human beings. With my work, I'm taking this concept one step further by introducing multi-being relationships, where the focus is not between humans and crocodiles as species and their relationships, but between individual crocodiles that the communities have identified over years of sharing space and resources with them. So a bit about the field sites where I'm working. This is in Western India, and I work in 4 different regions in Kutch, in Saurashtra, Central Gujarat, and South Gujarat. Today I'll be speaking mostly about my work from Central Gujarat. However, just to give a brief overview, I have selected these sites for a couple of reasons. So the site here is a place where crocodiles are ecologically present, but also present in the culture and the religion. So there are these temples, and people have religious association with crocodiles. This site in Kutch, crocodiles are ecologically present and their population is increasing. However, they are not present in the culture and the religion. And lastly, here where crocodiles have been locally extinct for about last 60 to 100 years. However, they still persist in the local religion and culture. So I'm trying to understand what drives human-crocodile relationships in these different socio-cultural and ecological contexts. But talking about Central Gujarat, it's interesting because these crocodiles are living in agricultural area, cities, towns, and villages. And here people are very fascinated by crocodiles. They share deep bonds with individual crocodiles, and the relationship that I would like to call as a positive relationship is something interesting that should be studied further. I'd like to give one example where there is also physical contact between humans and crocodiles, not in the form of attack or removal or retaliation, but when crocodiles enter the farmlands or places where they shouldn't be, people try to remove them, such as this. And just to clarify, these are not domesticated crocodiles. These are wild crocodiles. But they are quite stubborn, so then the farmers would give up. So these are the kind of relationships that I'm talking about when I say human-crocodile positive relationships, and which needs to be studied further and to understand why in certain places there are such kind of relationships and why in other places there are negative interactions. So for the first part of my work, I tried to understand the behavioral profiles of crocodiles in two adjoining regions of central Gujarat, one where there is more conflict, other where there are very few negative interactions. In both of these places, I selected sites which were quite similar in both population size, in terms of their interactions with humans. They were comparable at comparable distances from the nearest human settlements, and both of these sites of observation were just 40 kilometers apart from each other. I did instantaneous group scans to study crocodile behavior. Each scan was 30 minutes apart, and I did from 8 in the morning till 2 in the noon, and then 3 to 6. And during each scan, I recorded behavioral state based on the ethogram that we had prepared during a reconnaissance survey and their interactions with humans. So these are the, this is what we sort of end up with in our results, where on the right you can see the behavioral states that we could identify, and on the graph you can see the proportion of individuals observed in different behavioral states. Just to highlight a few important points here, that terrestrial movement was almost none in place where there are high negative interactions and where people prefer to remove crocodiles whenever they see them, whereas in other place there was quite a bit of terrestrial movement recorded. And our further analysis focused on the basking related behaviors, the surface, partial, and land basking, because these are the ones when they interact with humans the most. So we did further analysis using a pairs test, which accounts for two assumptions in behavioral data. First is non-independence of data, and second is extreme outliers. So here you can see that in places where there is very low negative interactions, crocodiles were observed basking on land much more, whereas in other place, which is just 40 kilometers away, where there are more negative interactions, they compensate not being able to bask on land by partial and surface basking. So there is clearly a behavior plasticity depending on their interactions and relationships with humans. Next, I wanted to understand how certain communities come to know other beings, especially crocodiles in this case, through their historically developed relationships with them. And for this, we are using the framework of vernacular ethologies, which is the local understanding of non-human behavior. And particularly in this case, vernacular ethologies are not just the understanding of behavior, but they also actively co-produce these relationships. So we are looking at it as a framework, not just as an approach. For this, we conducted long-term ethnographic fieldwork, conducted semi-structured and open-ended interviews with human participants, and data analysis included inductive coding using a grounded theory approach, which focused on the interpretive frameworks through which crocodile behavior was understood within the communities. For our analysis, we used the approach developed by Dominique Lestel and Florence Brunois called hybrid human-animal communities, essentially which talks about integrating ethnology with ethology and understanding these relationships as they are co-produced and not separately. We identified 4 key themes through our work, knowing individual crocodiles, where communities had, in different villages, had found different ways to identify individual crocodiles, and this had an important conservation implication because when there were negative interactions, they had issues with particular individuals and not with crocodiles as a species. And I would love to talk more about how they identify individuals and everything, but if there are any further questions about this, I would love to discuss this. But in the interest of time, I'm moving further. Second is anticipating danger from crocodiles. One of the most important things that came through our ethnography is that the negative interactions or the conflict incidents, the responsibility behind those incidents were distributed across environmental, social, ecological, cultural factors. It was not just attributed to crocodiles, as we have seen in many other places, but the responsibility was distributed, which had an overall positive impact on conservation outcomes outcome and species recovery in the region. We also recorded forms of interspecies communication where people would mimic crocodile hatchling calls to attract them towards them, and this activity is learned culturally from one generation to another, and this becomes a site for passing on traditional ecological knowledge about crocodiles and ways of coexistence that have been learned within the communities. And lastly, the ethical interpretation of harm and non-retaliation, where we are considering non-retaliation not as absence of action, but as an agentive action being performed by these communities, even in the face of negative interactions. And lastly, we developed a vernacular ethological processes model to understand how these different themes that we identified within the situational context impact long-term relationship with crocodiles. So starting from situated encounters to transmission of practices and ethical judgments, we tried to understand how can we map out these relationships and how does it really affect crocodiles and humans. Moving forward, we also suggest that in this case, vernacular ethologies can be constituted as a sense of coexistence ethics within these communities that is being learned and passed on from one generation to another. And we are currently documenting these vernacular ethologies and trying to understand how does that impact the coexistence ethics that communities have developed. Lastly, I would like to say that in a place like India, where crocodiles are increasingly coming into contact with humans, conservation futures will depend not only on practical measures, but also on understanding these relationships at a deeper level, and to also understand if there are positive relationships in places, then how do they work. And here we created a model to see how different methods from natural sciences and social sciences can be used to understand these relationships, and I would love to discuss this more with anyone who would like to talk about this. Thank you very much.
Thanks so much. Bang on time. It's really great to have all these students, postgrad students, doing studies of this human conflict and coexistence. So just a reminder that that Joshendu is a PhD student. Our next speaker is also a student, and we have a third student coming up. So it's looking good.
Do you want to announce Josh and while he's coming up?
Yeah. So Josh, do you want to come up? Joshua Barwick's a postgrad student at the University of South Wales, and his research focus is also human crocodile conflict mitigation.
Thanks. I've just heard from the organisers that tonight's dinner starts at 8 p.m, not 6 p.m, so we can all take a bit of a deep breath and the speakers can maybe speak a little bit more relaxed. What's wrong? My apologies. So now tonight is a paid event though. So if you did not register for this through your, when you registered on the portal and you want to go tonight, can you please go downstairs and make a payment? Thanks.
Right, yeah, exactly what I think. Just a bit of weirdness going on.
Okay, you can use this one.
I don't know.
Oh,
cool. Right, so get rid of this. Yeah, find yourself in there. There.
What's the right one?
Yeah. Just full screen it.
Ooh, how do I put lines here again?
You know what? I think we have to get rid of this one. Why is this not selected?
There's a way to do that.
Do you know how to do it? This is a PDF.
It's a PDF.
No.
Wait, it's not the right one. Oh no, it's not the right one. Is it the right one? go. Yeah, no, that's not the right one. I've got my flash drive, I can do it.
So why don't we do the, if you got it on a flash drive?
Yeah, just go back and grab it now.
Yeah.
Wait.
Right. He's going to bring his now, but I think it's a PDF, not a PowerPoint.
PDF?
Yeah. Can you do that? No. He's got it. Let's see what. So he's going to have to put it in on the memory pin. That's fine, yeah? Okay. Cool. And the next one after this is Zoom. Yeah, so I just click on the printer.
This one? Yeah, bottom one. PDF. PDF, okay. Take this one. Cool. Again. Okay. Cool. So I'll just tap through this as normal then. Cool. Nice. Right, hello, cool, finally getting going. So yeah, as you said, my name is Josh. I'm a, almost graduated master's student, and my interest is human wildlife conflict with crocodiles, but particularly from the social sciences aspect. So if we jump straight into it, I think my predecessors on the stage have kind of covered this enough for you, so I won't go into it too much detail, but a quick recap of why they're important. So things like traditional ecological knowledge inform us of different ways to mitigate conflict, like crocodile exclusion enclosures. Here, we were able to then come in and provide resources to develop more robust and strengthen their protection. And then if we go over to Sebastian Brackane's research over here on cultural importance, we were able to understand why existing mitigation was not working very well, and it was because of their cultural significance to the community, so we was able to work with them and develop a much more effective procedure. And then if you have a look here in the middle for necessity of community engagement, this is just a bit of a word cloud from a study that was interviewing local communities, and these trigger words were what they said in association with crocodiles and conflict. And not only does it cover things like their emotions and sentiments around them, but it's also got important things like there's location names down the bottom, habitat names like mangroves, it gives us important data there as well. And the current problem at the moment is that we kind of have a Western one-size-fits-all approach to it. So if I was to go get therapy, for example, I would want a licensed therapist, someone skilled, trained to do it, whereas we're all of us individuals, we don't have psychology degrees, some of you might, but we don't, and we're the ones dealing with these complex cultural situations. So that's kind of what we need to fix. Now, in order to look at this, I did a 25-year temporal literature review, not systematic, just a literature review, and I broke it up into 5-year segments, so 2000, 2005, and so on. And then I also sent out a survey to all of yourselves, where I was looking at your current knowledge on the subjects, your attitude towards them, do you use them, do you care about them in application in the field, and are you open to being educated further on them? Now, I would have liked to have walked you through the literature a little bit more, but due to time constraints, I'll give you a bit of a whirlwind view of it. So, as most of us probably know, crocodiles historically were hunted to almost extinction, and it wasn't until the 70s when legislation came in from the likes of Graham Webb here that they started to get a bit of population recovery. And then that started to happen roughly around the late 90s to early 2000s, which unfortunately then increased human-crocodile interactions and such for the conflict. So early 2000 literature was all conflict, how can we resolve this to stop the populations going back down due to the amount of conflict that was happening. And then as we go on to like 2005 to 2010, we started to recognize that there was some sort of relationship in the literature between the local communities and the culture and stuff and the crocodiles, but it was still very much seen as an obstacle to conservation. We didn't know much about them, we was kind of just still ignoring them and going on with our Western approach towards conservation. And then as we get towards 2010 to 2015, this was a really pivotal moment in the literature for crocodile conservation in the social aspect of things, because we were like, right, it's been 10 years now, and the conflict's not reducing, it's not working. what's going on, and that's when we started to actually look more into these relationships between the local communities, their cultural relevance, and just a general community engagement as well. And it started to be a lot more successful then. They were saying it's starting to see more results long term. And what also happened around this era, like 2011 onwards, you had the launch of different social medias that started to boom. So like ResearchGate, Twitter, and other social medias, they started to become a lot more popular, which means we had a lot more access to literature, publication, education, and also just media posts of where this conflict was happening and why. So then we was able to start putting a lot more research into it. And then as we get to 2015 to 2020, this was like a big push. It was like, right, local communities, culture, everyone pushing. We've got pioneering research such as that by Brandon Sidlow and Sebastian Brackane in Timor-Leste with the good grandfather crocodile, Lafayette, stuff like that. The literature started to notice there was actually a significant lack in education and teachings on these subjects as well. So we started to see the first curriculum being developed in schools around these subject areas. And then as we got to 2020 to 2025, it was like, okay, cool. Our conflict approach is not working. We know that it is essential to include our local communities in it. And we all know that conflict is not going to go away. It's always going to exist in some format. So the new goal is coexistence, as Simon Pooley will probably sing until the cows come home. So as we're going through that 2020 to 2025 literature, it's about shifting the narrative, then the perception. How can we go towards this coexistence goal? So it's changing human-crocodile interactions instead of always conflicts, and so forth. So yeah, the takeaway was basically that there's not enough education or teaching around it until the last 10 years. So that's very recent in the literature. Now, for my data analysis with a survey that I sent out to you all, you can take a picture if you want. These are the tests that I tried to do for both quantitative and qualitative analysis. And jumping straight into them, these are like research questions. So for this one, on the knowledge scores, I found out that the people who had formal education, as you can see by the middle ones here, actually scored higher. There was an average score of 3.33 across the board, but those who had formal teaching had a higher knowledge score as well. And then when I jump into sentiment, I found by professionals, so industry professionals and students, they had overall a very positive to neutral perception or attitude towards human-crocodile conflict in terms of traditional ecological culture, that kind of inclusion. So I was like, that's not really broad enough. So I looked at it on a sentence-level sentiment analysis, went into individual responses for every person there, and it found it was overall a very neutral. So 0 is neutral, below 0 is negative, past it's positive. So you can see there about a really high spike. Everyone had a really neutral response to why they're important and should be included, which for people that are meant to be rectifying these problems is not really what you want. You want a more positive solution. And then looking at their openness to further education, it had to rank the options, and the highest ranking ones were in-person workshops and in-person lectures. But the literature's already told me that there's actually a significant lack in the provision of those. So we're wondering why these things are lacking implementation in practice when there's just not the teachings around them. They haven't got the people that are skilled in training them actually running them. And then when you look at my qualitative analysis, I used like a deductive versus inductive hybrid method with like using the Braun and Clark method as an outline. And across the top there is the overarching themes that came out and the number of times they came out in the responses. And so from that, I created some deductive codes. So you've got the value in the relevant context, so that's traditional ecological knowledge, culture, community engagement, inclusion and exclusion dynamics, are they choosing to include or exclude them, why, the community role, so the role that the communities can play in this conflict mitigation, and then their stance on education, subcategories towards it, and then at the bottom is a couple of examples of the sort of words that the algorithm used to add those into those categories. Now, just to go into a couple of the responses, so as again, there was a statistically significant difference in learning pathway and their sentiment and attitude towards them. Now, across the top there, the formal professional, that's their learning pathway, and these quotes you can see across the middle are in response to a question on what does successful mitigation look like to them, to the individual. Now, I won't go through all of them because there's a million of them, but just to pick a couple out that stood out to me, a student who had had academic teaching, and then an industry professional over in the mixed section that had had academic and other teachings both came up with the same words. So people that have had the same sort of teachings are coming out with the same sort of opinions on what should be successful mitigation, whereas people who hadn't had any academic teaching, so those that just came from community, so learning from their peers, zero conflict. And as you are in the room will know, zero conflict is not possible. There's always going to be some level, but it's that coexistence again. It's what's an acceptable level of sacrifice on both sides. And now, just one quote that stood out to me a lot. Now, if you are in the room and I've misinterpreted this, you're more than welcome to come and talk to me. But this individual said they had no formal education on it, and that part of their job in the country is to work with the indigenous and the local elders and stuff like that in the mitigation process. And yet this was their sentiment of like the fauna belonged to the Crown, and that it was on private property, so they didn't believe they'd have to include locals in that instance, because their question is, have you ever chosen not to implement these in practice? So I just thought it was really interesting that someone whose job is to work with these would have that sort of analysis, but I won't go into it too much because it gets a little bit political there. Now this is just to give you a bit of an idea of the sort of demographics that did fill out my survey. So not a lot of people, only 30 of you filled it out, but it went quite around the world. Got 17 countries here that filled it out, so I thought it was pretty cool to get such a wide range of input onto the survey. Now, the future implications this has is on the screen here, and where I would like to go with it is to develop some sort of curriculum. Yep, cool. Some sort of school curriculum that can be offered out to different institutions, a workshop that can be delivered at events such as this, because obviously people said they want in-person workshops, in-person lectures. And then also the big one for me is collaboration. So if these situations like this, they're very context specific around the world. Every country you go to, it's very context specific of these cultural beliefs, their knowledge and stuff like that. And that's something that can't be taught. It has to be lived. So my solution to that is, if you look around the room, I would like all of you, if you just have a quick swivel, look around the room next to you. We've got a beautiful palette here of people with different knowledge, culture and community. So if we can work together, be less of just CSG members and be more of a CSG team, then we can start to tackle these collectively together rather than just, I've run a project, you've run a project. It is, we've run a project, and ultimately I feel like that will come to a much quicker solution to some of our coexistence problems. So just for our take-home messages here, coexistence is understudied because it was a heavy conflict focus for the first 10 to 15 years afterwards, and then the attitudes and perceptions and stuff are affected by learning pathway. There's that big lack of formal education in the literature, and people who are working in the industry are saying they want that formal education. And then the forward action is meetings like this, workshops, talking to people, working together. This is ECNM in Berlin. Thank you very much. Now, one second. Just very quickly, because I did, as you saw, I only got 30 responses on there. In order to produce accurate academic teachings, I am going to reopen my survey. So if you would like to either scan it there, or if you can't do QR codes, my email's up top there, and I can send it all out to you, and then I'm going to kind of redo this before publishing. Thank you.
Thanks. Well done. Where is this thing? So we have the missing mouse situation. There's Okay, so Annabelle, are you there? Annabelle, can you hear us? She has to take control. Annabelle? Okay, I'm not sure if Annabelle's, maybe she hasn't got the same time zone as we're on. She was here at the beginning of the session though. Oh, right, she's doing it. Great. Okay, Animal's on. Animal, we can't hear you. Hang on. Can you get the mouse back? It's on the bottom right. It's turned off. Yeah. Sorry, Animal. We're just getting the volume. Yeah. No volume. Can you speak, Animal? We're not hearing you.
Yes.
Great. Okay. Yeah. And go.
You can hear me now?
Yeah, perfect.
Thank you. Well, thank you for being able to be here at 1 o'clock in the morning on Gadigal country here in Australia. This presentation is hopefully brief, and it resulted from the Darwin CSG meeting where Gary Lindner and his team were present and talking about their on-country program to manage crocodiles in Kakadu. So, can't get rid of the bar at the top of my screen, so apologies if I'm not reading the titles out here. The project that Gary and his team undertake is to manage crocodiles in a very important area in the Northern Territory. And as we've heard, of course, the conflict arises when people do stupid people things and don't respect the crocodile in its natural environment, assume that there aren't crocodiles if they can't see them, when they undertake unsafe practices around boat ramps, and also when they encourage crocodiles to encourage interaction with crocodiles for photo opportunities and tourism thrills, and I guess most of all become complacent in crocodile country. Kakadu National Park is managed by the traditional owners of the land, and it's a very significant tourism and cultural place with boundaries along the East Alligator River, amongst others. And this river runs between Kakadu and Arnhem Land in the northeast part of the Northern Territory. The significant point is the bridge between these two lands, and it's called Carl's Crossing or Cahill's Crossing, and it bridges the land and is a tourist route between Kakadu and Arnhem Land. And anyone who's been on Facebook, particularly in Australia, will have seen the classic crossings at Cahill's. The tourists and locals rely on it because it's the point at which trucks and movement can occur. Fishermen take advantage of the tidal flow, which tends to concentrate the fish at the causeway when the tide's ebbing and flowing. And boaties use the nearby ramp to access a river that's not only rich in wildlife, but rich in fish as well. And this crossing is actually quite well signposted to say that there are crocodiles there, so if you're blind and you can't actually see the hundreds that line up to catch the fish, then there are certainly signs there that will indicate it's not a good place to be in the water. It's fairly self-explanatory why a management program is needed, and as you can see from the photos there, the area is significant and heavily populated by tourists and fishermen and the local people who use it for swimming and fishing and collection of other foods. The traditional owners of Kakadu, of course, have a deep cultural connection with crocodiles and their habitat, and they are very motivated to preserve crocodiles. The East Alligator program is quite significant because of its uniqueness in focusing on negative reinforcement of crocodiles by rangers to reduce interactions with human activity, basically to encourage crocodiles that being anywhere around people or boating activities is a really bad thing and that they should stay away. The animals that are targeted in this program are generally those that are over 2.5 metres and definitely those that are showing less fear of people, or as we like to say, are a little bit cheeky. Most of the animals in this program are simply caught, restrained with ropes for up to 24 hours, either at the site of capture or they might be removed to the local ranger station, and then they're released at the site of capture. And the purpose of this is simply to impress upon the crocodile that if they are coming anywhere near people, then something really unpleasant is going to happen. Not necessarily dangerous, of course, because there is crocodile welfare has to be considered, but that this is going to be an unpleasant experience and one which they don't want to repeat in the near future. Larger or more aggressive animals are caught, sedated and held out of water for a period of time, but then they're relocated to the more remote parts of the park where there isn't such, where tourism doesn't exist and where local people aren't inhabiting the waterways or utilising the waterways as heavily. And the purpose of this isn't to relocate the animal permanently. Crocodiles have a very good homing instinct, and so the negative reinforcement is not only the capture and the people where they can utilise the animal to share with the community, but also where the spirit of that animal can be released back into its natural habitat. And from a cultural perspective, this program run by the local traditional owners and rangers, giving complete cultural input from the people who live there, and also having a practical application of managing crocodiles that are dangerous or potentially dangerous to the public is really quite significant. So just briefly, the rangers have boats and they're all skilled in hunting these crocodiles. The target animal is harpooned through the dorsal cervical skin with a dedicated, specially made 4-pronged stainless steel harpoon head attached to a strong cord delivered manually through a harpoon pole. Crocodiles brought alongside, secured, and then hauled aboard or hauled alongside and taken back to the boat ramp for processing. It's quick, efficient and professional, and part of my purpose in being involved in this project was that Gary and his team had approached me after the last conference because they wanted a safety and welfare audit of the processes that they are undertaking to ensure that they were always going to be doing best practice and what was most humane for the crocodile, given that the purpose of this program was negative interaction and negative reinforcement. So we processed crocodiles in the middle of the night on a boat ramp in a river that's very heavily populated by very large saltwater crocodiles. It's an experience not for the faint-hearted, I can assure you. We tied the animals off to bollards so they couldn't escape back into the water while they were still partially restrained, partially drugged. I observed the process that was being undertaken, which was the rangers, in conjunction with a local fellow who's licensed to use tranquilisers and other sedation on crocodiles, and he provided the dose that he would normally use on these animals, and they were monitored and blood samples taken serially through the restraint process, and then they were released when they were safe to be released back to the water. So we estimated body weight from morphometric measurements, including total body length and snout to vent length, and I processed the samples looking at certain elements, particularly electrolytes, lactate, hematocrit and blood gases, venous blood, so there were limitations, to monitor what the welfare, physiological welfare on these animals was, and also to get an idea of the dose rates of drugs that are actually being used, because pancuronium and diazepam is a drug combination routinely used throughout Northern Australia to chemically restrain crocodiles in the wild, and the dose rates are very variable, as are the effects of the drugs. So retrospectively, it was determined that the dose rates of the pancuronium, which is the neuromuscular blocking agent, is so infinitesimally small that it probably wasn't having a lot of impact physiologically. I think the diazepam was probably having more effect. In spite of the low dose administered to the treatment group, most animals demonstrated reduced to absent reflexes for at least an hour, indicating that there was some effect of the drugs. And more significantly, the animals that had been administered drugs demonstrated significantly less acidosis, which is associated usually with stress and anaerobic metabolism. And all animals breathed voluntarily throughout the procedure at a rate of 1 to 4 breaths per minute. At higher doses of pancuronium, you often don't get voluntary respiration at that level, so which is very important to ensure good physiological response. The outcomes of this program, I think one of the most significant outcomes is that we have community and local people managing a problem or managing an issue so it doesn't become a problem, not only for their own people, but for the broader community, and that regardless of the outcome for the animal, cultural needs are being met, as well as keeping the public safe. It's probably one of the only non-lethal programs for cheeky crocodiles. The other advantages of this program is that it's educating people on their responsibility to their own country and all it contains, but more importantly, for outsiders coming in, it's educating them on the responsibility we all have to this place we call home, regardless of the colour of our skin or our heritage or our reasons for being in croc country, and engaging the traditional cultural wisdoms to balance safety and environment. It emphasises the need to be croc-wise in croc country to manage conflict rather than dealing with the consequences of a conflict. The chemical restraint options, part of the reason for doing this was because pancuronium is now becoming extremely difficult to obtain in Australia, and there need to be very effective and reliable and repeatable chemical means for handling large crocodiles in remote areas, particularly where you have minimal handlers with experience or just minimal bodies on the ground. And so the next focus of this research is going to be to look at other options of drug combinations that might be effective, and also from an ethical and welfare perspective to constantly ensure that catch and restrain protocols are being measured against good physiological welfare criteria to ensure best practice, and that this evidence will be published. And the benefits of this program, other programs like...
Annabelle, can you hear me? So we're kind of at time, so if you could conclude, please.
I'm sorry, I can't hear what you are saying, but just to wrap up, it's upskilling rangers, it's giving cultural ownership of these animals back to the locals, and it's also promoting good welfare and safety in this environment. Thank you.
Thanks to Annabelle. I should say, in retrospect, if you don't know her and looking at that scary work she was doing, she's a very experienced vet based at University of Sydney. Okay, so the next person, all of us who work in this kind of world know quite well, Brandon Sidlu. He's been working on these kind of issues for a long time, but he is currently formalizing this in a PhD study at Charles Darwin University in the top end in Australia. Brandon compiles the International Croc Attack database, Croc Attack, so, please send him your data. And he's a member of the Living with Crocodilians working group. So he's pre-recorded something, so let's see how this goes. I'm going to give us some volume. Yeah. So he's giving us 2 talks. The first is Indonesia. Let's see how this happens.
This presentation is on saltwater crocodile attacks in Indonesia.
Sorry.
Over the past decade, more saltwater crocodile attacks have been reported in Indonesia.
It's here. It's on screen, but it's...
Should I play it?
Yeah.
This presentation is on saltwater crocodile attacks in Indonesia. Over the past decade, more saltwater crocodile attacks have been reported in Indonesia than in any other country. Yet hotspots, victim demographics, and activity profiles remain poorly understood. To address these gaps, we compiled the records of all saltwater crocodile attacks reported in Indonesia between 2016 and 2025. All incidents were sourced from the Croc Attack database, which is at crocattack.org, which I compiled over the past decade from a combination of fieldwork, news media articles, and correspondence with colleagues, government officials, and NGOs. Minimum data requirements for inclusion were attack location, outcome, which is fatal or non-fatal, and year. When available, we also included victim age and sex, activity at the time of attack, and the exact date. For the analysis of spatial trends in attacks, we grouped attacks by both province and region, namely Kalimantan, Sumatra, Sulawesi, the Lesser Sunda Islands, the Maluku Islands, Java, and Papua. We grouped victim activities at the time of attack into 4 broader categories: fishing-related activities, activities related to domestic and subsistence water use, non-fish resource harvesting, such as sago and oil palm harvesting, and an other category for activities less commonly reported, such as leisure and traversal-related activities. For the 2016 to 2025 period, we recorded 1,217 saltwater crocodile attacks, including 588 deaths. Attacks were recorded in 31 of Indonesia's 38 provinces. No attacks were recorded in Bali, Central Java, East Java, Jakarta, Yogyakarta, Highland Papua, or South Papua provinces. Indonesia experienced at least 21 times the number of attacks and 49 times the number of fatalities reported in Australia over the same period, despite likely harboring only a fraction of the crocodile population. The highest numbers of attacks were reported in Riau, East Kalimantan, East Nusa Tenggara, Bangka Belitung, North Maluku, and Southeast Sulawesi. These 6 provinces accounted for 50% of all attacks reported in Indonesia. These provinces do not share any obvious set of characteristics that would distinguish them from the remaining provinces where attacks were also recorded. Moreover, 17 of the 31 affected provinces reported more than 30 attacks, indicating this is a widespread issue in Indonesia. Both fatal and non-fatal attacks increased significantly over the decade period at the national level, while increases were also noted for 11 of these 31 provinces. The reason for this increased attack frequency is likely multifactorial and may vary between regions. Crocodile population recovery is among the most frequently cited drivers of increasing attack rates in other range states and likely contributes to patterns observed in Indonesia. However, the extent of this recovery remains unclear. Surveys at attack hotspots in East Nusa Tenggara province revealed very low non-hatchling densities, and similarly low densities have been reported from parts of Bangka Belitung and Sumatra. Since high attack frequencies can occur despite low population density, suggests that factors other than crocodile numbers are driving conflict in at least some of these regions, though population status remains unknown across most of Indonesia. Another factor that may have contributed to increased reports of attacks is increased access to the internet and social media in Indonesia over the past decade, particularly in remote areas. In 2016, only 25% of individuals had access to the internet, but by 2024, this had increased to 73%. Human population encroachment and certain industries have been implicated as indirect drivers of increased attack frequency in many areas. Tin mining in Bangka Belitung creates excavation craters that subsequently flood, forming water-filled ponds known as kulongs that can connect to adjacent waterways during high water, enabling crocodiles to enter areas usually beyond their range. A structurally analogous mechanism has been proposed for oil palm plantations, where extensive canal networks expand crocodile access into previously uninhabited areas. Nickel mining pollution in central and southeast Sulawesi is believed by local communities to have resulted in more crocodile attacks due to the depletion of fish populations. In East Kalimantan, habitat loss, pollution, and prey depletion linked to the development of the new capital Nusantara has also been implicated in increased numbers of saltwater crocodile attacks. Most of the victims in Indonesia were male, and most were aged 31 to 50 years, the prime working years. Though children aged 1 to 10 years comprised only 5% of victims, they experienced the highest fatality rate at 70%. This is consistent with their smaller body size, thus expanding the size of crocodiles that can actually successfully prey upon them. At the national level, most victims were engaging in fishing-related activities, followed by those related to domestic and subsistence water use and non-fish resource harvesting. Notable deviations from the national baseline were observed at the regional level. In Kalimantan, more victims were attacked while engaging in domestic water use activities than fishing, while non-fish resource harvesting activities were much more common in Sumatra. Fishing-related activities dominated, however, in the Lesser Sunda Islands and the Maluku Islands. Female victims were significantly overrepresented in several provinces that also exhibited elevated domestic water use incident rates, most notably in Kalimantan and Sumatra. This pattern likely reflects traditional gender roles shaping both activity class distributions and provincial sex ratios in areas with more extensive freshwater habitat. The most frequently reported non-fish resources being harvested at the time of attack were tin, all of which occurred in Bangka Belitung, sago, all of which occurred in Riau, timber, and Kangkung. All victims harvesting tin, sago, and timber were male. The distribution of victim activity classes strongly implicates direct waterway dependency as the primary driver of attacks, with fishing and domestic water use activities collectively accounting for 78% of incidents. Domestic water use related attacks likely reflect the absence of piped or pumped water access, compelling residents to use natural waterways for bathing, domestic tasks, and sanitation, while fishing in Indonesia is overwhelmingly livelihood or subsistence based. Provincial variation in activity class distributions likely reflects regional differences in waterway dependency. Fishing-related incidents were overrepresented predominantly in the island provinces, East Nusa Tenggara, Maluku, and North Maluku, where freshwater habitat is less extensive than in the Greater Sunda region. Incidents related to domestic water use, on the other hand, were overrepresented in Kalimantan and Sumatra, where there are large tidal rivers that extend hundreds of kilometers inland with extensive freshwater wetlands. The overrepresentation of non-fish resource harvesting in Sumatra is reflective of the sago and tin mining industries in Riau and Bangka Belitung, respectively. Assuming the continued viability of the saltwater crocodile population in their present range is a priority, effective and sustainable management planning requires baseline population data. Surveys should therefore be prioritized in high conflict areas and representative high quality habitat sites within each province, rather than attempting a countrywide assessment, which would be impractical given Indonesia's size. Surveys must employ consistent methodology conducted by trained personnel reporting non-hatchling densities. In Kalimantan and Sumatra, where saltwater crocodile range overlaps with the Sundagarial, and in New Guinea, where overlap occurs with the New Guinea freshwater crocodile, surveyors must also be competent in species identification, which has proven to be an issue in some recent surveys. The considerable regional heterogeneity across Indonesia precludes a uniform management approach. Provincial level strategies should account for crocodile and human population densities, habitat quality and quantity, long-term habitat security, and local cultural beliefs regarding crocodiles. Mitigation strategies should include crocodile education programs, similar to the Crocwise campaign in Australia, the provision of alternative water sources to communities lacking safe access to fresh water, construction of crocodile exclusion enclosures and other barriers, sustainable crocodile management dependent on provincial crocodile population status, and pressure on multinational industries, such as tin mining and palm oil, to increase regulations and provide workers with safe places to bathe and fish. Thank you.
Okay, thanks to Brandon. I'm going to call on my Batman here. Thanks, Ashende. So we have another recorded talk. Uman. By Mira Anna Uman.
Hello, everyone.
Yeah.
Greetings from the Andaman Islands.
Could make it a bit louder, maybe slowly. Okay, we're nearly there, guys. There's 2 to go. 2 talks to go. So Mira Anna Uman is founding director of the Dakshin Foundation. And you okay? Yeah.
Apologies for not being able to join in person this time. And if you're seeing the recorded version of the talk, it's because there has been a lack of internet connectivity in this region, either due to the weather or power outages and so on. And I've been trying to record this talk without all the birdsong and the cicadas buzzing in the background, but I'm afraid they're not stopping today. So I hope you can hear me. Anyway, today I will try and give a very brief introduction to various aspects of our work on the interactions between people and saltwater crocodiles in these enigmatic islands. As you may already know, the Andamans are an island group which are a part of India, but are located in the Bay of Bengal about 1,300 odd kilometers east of mainland India, and several hundred kilometers west from the coast of Myanmar. When Zai and Rom Whitaker and colleagues at the Madras Crocodile Bank carried out the first surveys here in the 1970s, they discovered that the saltwater crocodile numbers, as was the case for the rest of India, had been depleted considerably here as well. Since then, over a period of time, croc numbers have increased, and over a period of several decades, there have been a variety of drivers of population recovery, at least what we think are the factors. This includes first and foremost the enactment of the Indian Wildlife Protection Act starting in 1972, which eventually, with stricter implementation through the 1980s and the 1990s, brought in a range of conservation measures, including a ban on killing or using the species in any way, and also enhanced habitat protection. The tsunami following the Sumatra earthquake of December 2004 also likely increased available waterlogged habitats for crocodiles. And finally, starting with the early 2000s, Increased military surveillance for this strategically important region is believed to have reduced the removal of saltwater crocodiles, both by local Indian citizens as well as by foreigners coming in from places like Indonesia, Burma and Thailand to harvest crocodiles. In recent years, these factors have kind of collectively contributed to an increasing population of crocs, so much so that conflict with crocodiles have become a major issue in the islands, which affects both local livelihoods and also conservation success. So the work that we are carrying out currently is towards understanding various facets of this problem, and we are working towards improving outcomes for both people and crocodiles in the face of conflict, which has been increasing. So on behalf of my co-authors and advisors, Ram Whitaker and Graham Webb, who I hope would have joined today either in person or remotely, I will run you through a quick summary of what we've been doing. Most of the work that is being done currently is with a view to gain knowledge or to inform certain aspects of human-wildlife conflict, human-crocodile conflict, through applied research. We are also in the process of rolling out a series of interventions. In the last few years, we've been working on recording and mapping attacks in the region, understanding risks to humans and livestock, developing different models for interactions between people and crocodiles, and also developing a model of the system of conflict as a whole, understanding fear and stress in relation to wildlife attacks, providing best practice guidelines, safety instructions, and also awareness generation for different target groups. And most recently, our intervention work has also been focusing on dealing with poultry, abattoir, and fish waste, as their disposal is usually in mangroves and creeks, and that tends to attract crocs. Most of our work of this nature has been in the South Andaman landscape, where which also has the maximum human population in the Andamans. Other aspects of our research and interventions also take into account the Andamans as a whole, or even the neighboring Nicobar Islands. In South Andaman alone, we recorded and mapped about 45 attacks that have taken place since the early 90s. We are also mapping significant habitats such as mangroves, recording interviews with survivors and the families of victims. We visited almost all settlements, which are about 54 settlements in this region, and found that there were at least twice as many cases of croc attacks as were reported in official records, or even captured in earlier databases such as Crocbite. And this actually reflects the fact that in the case of smaller non-fatal incidents, there's a reluctance to report this by the community, as they fear that they can get into trouble with the law, because many of these attacks take place in protected areas, especially mangroves and creeks, which is supposed to be out of bounds for local community members. Overall, the trend in increase in conflict has been gradual, but an interesting pattern is that there were only 4 or 5 attacks before the year 2000, and the majority of them have taken place after, which reflects perhaps both increasing populations of humans as well as crocodiles in South Andaman. We use both existing information from available data sets to understand generic patterns related to risk, and also looked at global information and looked for comparisons, that is similarities and differences between the Andamans and other sites. An initial broad-based risk map has also been developed based on our knowledge of ecology and behavior of salties, along the lines of the habitat map that was prepared for East Timor. However, I wanted to add that this is a work in progress that needs to be, the map needs to be specified further, and it also remains to be validated with, actual data on crocodile presence. We are in the process of working with computational data scientists to build local agent-based models for understanding croc population dynamics, as well as interactions between crocodiles and human agents. This is a work in progress. Similarly, another piece of modeling that is in progress relates to the modeling of conflict using a wicked problem framework. The attempt here is to build what is known as an order of magnitude model of the system as a whole, which interact with conflict, and to find ways to represent or to visualize these interconnections, and hopefully modeling predictive what-if scenarios at a future date when we have sufficient information to populate the model itself. I want to add here that both these forms of models are of an extremely exploratory nature, and Graham and I have had several discussions about how far some of this might actually work. So to be completely frank, it remains to be seen if anything meaningful, or rather I should say if anything, any practical insights can be gathered from them eventually. Our work on crocodile attacks, especially interviews with the survivors of croc attacks, is also informing a couple of upcoming publications of on human fear of predators, risk aversion in people, resistance to conservation, and so on. And from time to time, we've produced some amount of literature relating to safety guidelines, best practices, and so on, which are given to government agencies. A recent large project that we've started focuses significantly on waste generation and its management, especially in mangroves and creeks. This is so as to reduce the interface between crocodiles and people in these habitats. This particularly relates to the dynamics of disposal of poultry, fish, and abattoir waste in the landscape. Currently, we are in the process of mapping and identifying hotspots of disposal, and the idea is to repurpose this waste for other purposes, hopefully by the creation of circular economy pathways, say for instance taking these forms of waste and supplying them to piggeries, fish farms, and so on. As is the case of successful conservation in places such as Northern Territory in Australia, we also aim to experiment with monetary value creation as well as revitalizing cultural threads. This project also aims to develop a more appropriate compensation design, and the whole process of receiving compensation from the government has been a problematic one in the islands due to a number of reasons. Another component of the project has been to develop better outreach and awareness material for different target groups, then also to hold and facilitate multi-stakeholder consultations to aid in decision making towards better outcomes for both people and crocodiles. And most of this is work in progress, and I hope to come back again with more results and outcomes. But what needs to be remembered is that a number of challenges remain for resolving conflict in places such as the Andamans. Despite having a diversity of human-animal relationships ranging from protection, worship, lethal control, use, and also ambivalence, all of which signals possibilities for different coexistence-centered strategies. India is also well known for its often inexplicable adherence to fortress conservation measures by the government departments and by the majority of conservationists. There is a strong protected area network which excludes different communities in the islands. These are the settler communities who are the already marginalized groups, and convincing the state to look into measures such as sustainable use of crocodiles, or for that matter any species, has been largely unsuccessful. And this has resulted in increasing conflict, retribution and resistance by local communities who are now opposed not only just to having crocodiles in their backyards, but also end up being opposed to conservation in general. It has been an uphill task to get both communities as well as government agencies to the discussion table even. And in the Andamans especially, these two groups are quite polarized in the way they are positioned. So a large part of our recent work with the communities as well as with the fisheries and forest departments are primarily aimed at building capacities, exploring improved livelihood solutions, and bringing these groups together to hopefully agree on a pragmatic middle ground. And this middle ground, of course, will entail some removal of crocodiles, hopefully some sustainable use, but primarily the creation of material as well as cultural value for saltwater crocodiles. So this is a brief summary of our work in the islands, and much of it is ongoing. I wanted to give a shout out to several team members as well as collaborators and advisors who have been involved in various aspects of this project. And if there's an internet connection, I will be there to take questions. If not, do reach out an email and I will respond to you to any specific queries. Thank you for this opportunity to present.
Great. So
my presentation is on saltwater.
Yeah. Brandon's on a mission. So we have Brandon's stereo. Brandon, final talk. You'll be encouraged to know, but this should be interesting. He's talking about Papua New Guinea, and it's been a huge blank. We know fascinating things go on in Papua New Guinea, culturally and otherwise. My dad was there with Federico Medem in 1976 for the CSG, trying to figure out if community croc farming was a thing. That's another story, a long story, an ongoing story. It's a tricky place, and so Brandon is bravely including it in his PhD.
Crocodile attacks in Papua New Guinea from 2015 to 2024. Two crocodile species are native to Papua New Guinea: the New Guinea freshwater crocodile, which is endemic to the island, and the wide-ranging saltwater crocodile. Despite their common name, saltwater crocodiles are often found at high densities in freshwater habitat. In PNG, they can be found up to the Indonesian border along the Sepik River and through Lake Murray and the Strickland River along the Fly River drainage. They are also found in suitable habitat throughout the island provinces. Sporadic reports in the news media suggest crocodile attacks are a frequent occurrence in PNG, but exact numbers and details have been difficult to come by. Most attacks are assumed to go unreported due to the remoteness of affected areas and very limited police and medical infrastructure. To determine the frequency and characteristics of saltwater crocodile attacks across PNG from 2015 to 2024, we conducted a countrywide survey collecting data from all provinces in which the species is believed to occur. Saltwater crocodiles are likely present in 14 of PNG's 22 provinces. Field interviews were conducted in 79 villages across 5 provinces, 11 in Central Province, 37 in East Sepik Province, 4 in Manus Province, 16 in West New Britain Province, and 11 in Western Province. The remaining 9 provinces and remote areas of the surveyed 5 were covered through phone and email interviews with residents. To be recorded, an incident required at minimum the approximate year, location, and outcome, which is death or injury. Preferred but non-mandatory data included victim sex, age, and activity. Records were then cross-referenced with the Crocodile database to avoid duplication. We collected the details of 584 attacks, resulting in 369 deaths that reportedly occurred during the 2015 to 2024 study period. Only 6% of incidents had been reported by the news media, confirming widespread underreporting. Records came from all 14 suspected saltwater crocodile inhabited provinces. The highest numbers came from East Sepik, West New Britain, Gulf, and Western provinces. Together, these provinces accounted for 69% of all reported attacks. Nationally, most victims were male, but females experienced a higher fatality rate. The proportion of male victims ranged from nearly all in Sandown, New Ireland, and Central to roughly equal with female victims in Gulf, Western, and East Sepik. Most victims were engaged in fishing-related activities, followed by those related to deficiencies in water, sanitation, and hygiene infrastructure, also known as WASH. Fishing was more common among men, while WASH-related activities were overrepresented among women. WASH-related activities were more common in Gulf Province, West New Britain Province, and Morobe Province, while fishing was more common in Central, Milne Bay, and New Ireland provinces. In East Sepik, a relatively high number of victims, all male, were attacked while hunting crocodiles. We detected no significant change in fatal or non-fatal attacks at the national level over the decade. The only significant provincial trend was an increase in non-fatal attacks in East Sepik. The high attack numbers in East Sepik, West New Britain, Gulf, and Western provinces are likely attributable to several factors. Fieldwork was conducted in 3 of these, and a hospital supplied much of the data in Gulf Province, likely contributing to more comprehensive records. East Sepik, Gulf, and Western are also among PNG's largest provinces, with extensive, suitable habitat. The northern coast of West New Britain also holds significant estuarine and freshwater habitat, though much of it is now dominated by oil palm plantations. Female overrepresentation in WASH-related attacks likely reflects the disproportionate burden of domestic water use borne by women under prevailing gender norms. The high proportion of victims attacked while hunting crocodiles in East Sepik is consistent with the region's culture and sustainable use statistics, with the Sepik River accounting for over 50% of national skin exports. Direct dependency on waterways for subsistence fishing and basic domestic needs is the dominant factor contributing to attacks in PNG. This is consistent with other low and middle income countries where livelihood-driven exposure has been widely identified as the principal driver. It contrasts sharply with high-income countries where attacks more commonly result from recreational activities. In Australia, for example, victims are typically engaged in swimming, recreational fishing, or tourism, rather than activities necessary for survival. The contribution of population recovery to attack frequency remains unclear, as the saltwater crocodile population outside of the Middle Sepik has not been assessed since the early 1980s. Limited historical data indicates that attacks were frequent even during the heavy commercial hunting period of the 1950s and 60s. In addition, a 1995 CSG newsletter report from West New Britain even attributed at least 12 fatalities to a single crocodile. Furthermore, depletion and recovery are also likely to have varied considerably between regions. Any mitigation strategy for PNG must account for the country's logistical difficulties. Attacks are not an issue in the major population centers of Port Moresby and Lae, and are instead concentrated across hundreds of remote villages, many reachable only by boat. Fuel cost, particularly in remote provinces, is prohibitively high, constraining the operational reach of management. An approach similar to what is effective here in the Northern Territory, in which problem crocodiles are consistently removed from areas of human concentration, is therefore unlikely to be a realistic option in PNG. The same constraints that limit management also limit hunting pressure, which has likely contributed to the persistence of saltwater crocodile populations in some of these areas. In Western Province, crocodiles are routinely killed on site whenever the means are available, and we were shown numerous photos of large crocodiles killed along the Fly River in recent months, with some skins destined to buyers in Indonesia despite exceeding the legal export size limits. A structured training program could redirect this practice towards targeted removal of problem crocodiles, though monitoring for safety and sustainability would be difficult. Up to a third of attacks could be prevented through wash infrastructure, such as piped water and water pumps. Crocodile exclusion enclosures, or CEEs, placed strategically along waterways could further mitigate wash-related incidents, and discussions with people in East Sepik indicated clear local support. However, all such infrastructure requires regular maintenance, which, given the logistical constraints, would likely need to be carried out by the community members themselves. A crocodile safety education program modeled on Australia's Crocwise campaign is a realistic and cost-effective approach, though its efficacy is likely to be more limited in PNG, where many people have no choice but to use crocodile-inhabited waterways for their domestic and livelihood needs. However, every community we visited was supportive of crocodile education programs. Our data relied heavily upon the recollection of residents. This method of data collection has some very important limitations. Crocodile involvement in unwitnessed fatal attacks cannot be confirmed, and recall accuracy varies, particularly regarding the incident year. These factors may partly explain anomalies, such as the disproportionately high attack frequency and fatality rate in West New Britain. In addition, while most attacks likely involve saltwater crocodiles, we cannot rule out occasional involvement of New Guinea freshwater crocodiles, particularly for non-fatal attacks far inland. A multifaceted approach is likely needed. Improved wash infrastructure, protective barriers, including fences and CEEs, crocodile education programs, and the targeted management of problem crocodiles. Given the lack of crocodile population data, any management plan should be preceded by efforts to better understand crocodile status and distribution outside of the Middle Sepik, using spotlight surveys where applicable or aerial nest counts. This should be done in order to ensure the sustainability of any management plan. Thank you.
Great. So with that, we conclude the talks. And could I invite the speakers that are here to come up and take a pew? Don't know if does this work. Don't know if any of these are working. Hello, hello. Hello, hello. Yeah. Josh. Okay, and I think we may have Annabelle still online as well. So I don't think we have Brandon or Mira available for answering questions, but if you have questions for any of these speakers, yeah, at the back there. Is there another? No. Hello, hello. Is there another speaker somewhere? I mean, another microphone. No. Oh. John, do you mind? Sorry. There's one there, but do you want to just take this one? Yeah. Is there a question in the meantime?
There we go.
Oh, great.
Okay, so first, obviously, great talks. I won't play favourites and say which one was my favourite. And I know that a lot of these issues that we face with crocodile-human conflict are going to have multiple solutions. In fact, even single problems could have multiple solutions. So this question is more of a sort of like brainstorming question on one particular issue. And I've got a few ideas of potential solutions myself. But it's on the more societal issue that we have in the modern day regarding social media, and the fact that we've got all of these, you know, clout chasers and that will go out for the photos and all that with animals, grabbing crocodiles and such, or snakes, etc. And then, of course, that encourages others to do the same in the hopes that they'll go viral or whatever. So it's more, do we have any specific ideas to tackle that problem outside of maybe some of the others?
If I may, so one of Bob Irwin Senior's legacy piece of work that we are committed and continuing to advocate and agitate heavily for is encouraging and demanding that the government actually regulates their own laws. So in Queensland and across all of the states and territories in Australia have very similar wildlife protection laws. It is illegal to interfere with a protected animal, and interfere means taking, touching, luring, hunting, you name it. What we're seeing is this media sensationalisation and monetisation of crocodiles and other animals across Australia, and our governments are not doing anything about it. They're saying to us that, oh, we can't tell the jurisdiction. Well, we say you can actually tell the jurisdiction. I've worked previously in private law, and where they have collected laptops, they've searched for data, and they've prosecuted. So I think we have a real lack of actual boots on the ground and number of people in the regulatory space actually taking up this work. We think it's really important because what we're seeing is that the more people are doing it and monetising it, that, like Alistair McPhee that I was talking about, he could not be prosecuted, the government said. Now, he knew that crocodile was there. They said that they couldn't prove his intention because he was an ambush predator. So that's why we went through the legal changes and the policy work to actually try and overcome that couldn't prove intention. Here we're seeing activities where people are deliberately interfering and interacting with wildlife, putting it on social media, becoming famous and encouraging others to do so. We would have seen the American with the wombat, baby wombat on the side of the road in South Australia, picking it up, screaming, Tarzan. He did it in the Northern Territory and in Queensland, and we're not getting that regulatory control. So we are working really, really heavily and advocating and pushing against the government across Australia, but particularly I am Queensland focused on holding them to account to make them do the job that they are legally required to do. So it is policy and it is legal. We have the laws in Australia to manage this. We just need our governments to do their jobs.
Just to give a quick input for an idea I would have as well, so as well as obviously legal policy enforcement, I would also say working with these social media influencers as well. So at the end of the day, there's always going to be people that want this attention, they want that social media reach, they want those viral videos. So if we can communicate with them and work a way that puts our message across of safety, staying away from them, they can still get the same level of engagement on their videos, but in a much more safe and effective way rather than promoting unsafe actions. Or
Yeah, I was actually
well.
going to suggest that apart from maybe making some of the punishments heftier, to actually have our own social media campaigns to kind of counteract it, fight fire with fire, as it were.
Yeah, do you want to add something? I just had to add one small point that, as you said, that it's very contextual, and something, a different perspective that I would like to put here is that, as you mentioned, even in India we have a Wildlife Protection Act that protects crocodiles, and nobody can come into physical contact with them for any reason, and it's punishable. Oh, sorry. It's a punishable offense, and people do go to jail for that. But in certain places where there is a positive relationship, where people don't really mind crocodiles, as you saw in the video, they were trying to remove crocodiles. Sometimes when they get stuck in the fishing nets, they hold them by their hands and remove them. So they are not coming into this physical contact to harm crocodiles or to get a social media fame out of it, but it's part of their daily routine that has been going on for generations. So sometimes this creates a conflict between governments and these communities, because there is no really conflict between people and crocodiles here, but that creates a conflict. This relationship creates a conflict between government and the communities. So, yeah, exactly, yeah. So in some of the places where I work, the NGOs, they work closely with the communities, to make sure that if someone from the outside is coming and touching these crocodiles or harassing them, then sort of register a complaint with the forest department, and also working with the forest department that these people are not really harming crocodiles. It's the people who are coming from cities or who are watching these videos online and coming here to see crocodiles close by. That's what creating problem. Yeah.
Okay, another question here. Graham in the front here. Oh, okay. So we'll go there, and then can we have Graham after?
Okay,
yep.
great.